Okay. Hello, everyone. My name is Leena AlZayer. I'm a second year medical student with you guys doing neuro. And today we'll be doing an overview of the neuro OSPE. Okay. So it's going to be kind of heavy. I will have to be كذا honest with you. It's a lot. بس أنا من النوع اللي I can't keep any details. So I am a very detailed explainer, and every time I do a question or a practice question, I go back to the concept and kind of revise it. So bear with me if I take a bit longer, but inshallah it's going to be a good revision for not only OSPE but also some SAQ and MCQ. فإنشالله it helps. I'm sorry from now إذا صار في any gaps in my information, any mistakes, please stop me and correct me. يعني I tried my best بس لعل يعني يكون في غلط وأنا ما انتبهت له please let me know and keep me posted. It's very hard for me to go in the chat, so I'd prefer you unmute to answer questions or have any questions. بس يعني if you don't feel comfortable, please send the in the comment place thing. And yeah, I think we should get ready to start. بس كذا حبيت أحط آية في البداية عشان أذكركم إني إنسان ويمكن أغلط، ومعليش اعذروني إذا غلط. بس إن شاء الله يا رب يعني I tried my best to do well in this presentation. So بسم الله, let's start. Okay, so in terms of what we're going to be doing today, we're going to be going over 6 labs for anatomy, 1 lab for micro, and 2 labs for physio. So my approach so the labs are a bit confusing and they have a lot of missing information from the lectures. So I add a lot from lectures, but then there's still يعني, it seems like the information never ends. So I also couldn't put every single lesion of the brainstem. I couldn't put every single lesion of the spinal cord, but unfortunately, it does come in the OSPE. So I tried my best to see what's high yield. In terms of physiology, it's the only one where I don't necessarily explain the lab because the labs are very, يعني, you guys know how physiology labs are. They're kind of يعني pointless. pointless, بس يعني I did kind of bring a question and explain how to answer it. And that's kind of my approach for physiology. But the rest, I do explain what was mentioned in the lab and then do questions, if that makes sense. And it's very, very similar to how I approached POD's OSPE review. Okay, but I just wanted to say a disclaimer, and I didn't do this alone. I used a lot of references from the summaries of our batch. I used lab committee stuff, screenshots from some notes from MYA, from Marwa, and a lot of other notes I get from the group chat, because you know how condensed neuro is. There's basically impossible for me to do it all myself. So I just want to give a shout out to them. يعني study smart, not hard. Take the information from people, and inshallah you will benefit. So let's go ahead and start. So the first lab was the organization of the nervous system. And in the beginning of the block, we kind of didn't know all these cross sections they just bombarded us with. And now that we are at the end of the block, we can have this overview and we kind of know, oh, what the thalamus is, what the internal capsule is. When the first lab, we were just like, what is all of this? So the organization of the nervous system is kind of the longest part of this OSPE review, because now I'm going to actually go in depth to every single thing we studied that we didn't understand in the first week of lab. So let's start with the organization of the nervous system from a histological point of view. So first, we just have a regular staining of hemolytoxin and eosin, or H and E stain, and we can see just a normal neuron. So what we can see here is a slide of as I said, H and E basic staining, and we can see these pyramidal neuron cells, and we can see these are the cell bodies, and inside the cell body, you can see the nucleus and the nucleolus. Around it, you can see the nisal body, so you can see so nisal body represents availability of ribosomes, and obviously, as we know, ribosomes are available in the cell body of the neuron. So this indicates that we are at the location of the cell body of the neuron. So a very good indicative thing that you are in the cell body is that you can see these nisal bodies, these dotted structures. So this is the perspective in neuron. What about the peripheral nervous system, where you just have axons and not the cell body? So this is a bunch of axons, and the way you can really differentiate between them is that you can see that dot, and in the middle, and then around it is that white area. That white area is actually the myelin sheath of the axon. So as we can see here, as I'm pointing with the pointer over here, in the middle, we have the cell body, or the axon, sorry, not the cell body, the axon, and around it is the myelin sheath, indicated with the letter MS for myelinated sheath. The pink reddish stains over here are just blood capillaries. So anytime you see these, you can also see here another one, and please follow the pointer with me. And over here, you can see all these capillaries, these red strands. But these kind of fried egg appearance, where you have a dot in the middle and around it is white, that's an axon and a myelin sheath around it. And again, I just want to reiterate, this is a neuron, and we have the nissel bodies around it in the cytoplasm. This is another picture of a neuron where you have the dendrites all over, and you have the axon going all the way to the terminal synaptic terminal. These dots around the neuron, you can say, are the supporting cells. It says my internet is unstable. Am I cutting off? Just please let me know if I'm cutting off in the middle. Can
You were
everyone hear me?
بس بعدين you're good. You're good
Okay.
now.
Okay, these dots here in the middle are basically supporting cells. They're glial cells of the nervous system, and they basically support and help the neuron to get nourished and so on. This is a slide from another perspective of the myelin sheath. It's just using silver stain, and basically we can see the myelinated axons here. You see these fried egg appearance where you have the black dot in the middle and around it is white. So I just wanted you guys to notice that this is the myelinated axons. And in the middle, where you have kind of this head, you can see the pyramidal cells, pyramidal neuron, sorry. So this would be a zoom of a bit of the gray matter and a bit of the white matter. The white matter being the axons with the myelin sheath, the gray matter being the pyramidal cell neurons over here. All clear? Any questions till now? Okay, please talk to me if you have questions, feel free. This is another picture. As we said, these kind of triangular shapes are the neurons. The dotted shapes around it are all glial cells that support the neuron's environment, and any strands that are kind of pink and long are these capillaries. So this is a capillary, this is a capillary, this is a capillary. These pyramidal shaped cells are neurons. They kind of look like triangles, and the neuropils and the glial cells are just the dots. The glial cells are the actual cell bodies of the supporting cells, and the neuropil is basically a bunch of axons and dendrites kind of mushed up together, and they make up the rest of the space of the nervous system. So these are the type of stains that they didn't mention in lab, actually, but I think that it's in Vlad's lecture, Dr. Vlad's lecture, and I think he kind of likes these questions even in an MCQ, asking, for example, what type of method is this? What can you know? What can you infer from this type of stain? So in the first one, you have the Golgi method, or the reduced silver method. Over here in this type of staining, on the Golgi method staining or the reduced silver method staining, you can only identify branching patterns. You cannot see cell body, you cannot see ribosomes, you cannot see nissel, you cannot see anything except the branching patterns of the dendrites and the axon. Okay, so this is the Golgi method. Then you have the Nissel method. The Nissel method is basically detecting the Nissel bodies, which are ribosomes, and it's basically a basophilic stain, but because you're focusing on these ribosomes that are in the cell body, in the cytoplasm, you are able to see the neuronal cell bodies specifically. So this way you can count how many cell bodies you have in a slide, and you can also count how many the size of the actual neuron. So if it's big, if it's small, and how many you have. So the Nissl method is helping for the size and the number of the cell bodies by seeing its ribosome/Nissl body substance. Then you have the Weyert's method stain or Weyert's method stain, which basically colors the myelinated axons black, and the gray matter that's actually cell bodies that isn't myelinated uh, doesn't stain it. So the black stain of the Weyert's method stains the myelinated axon. So as we can see, this is a section of the spinal cord, and the color black over here is all just a bunch of axons and fibers passing through the spinal cord, while the middle, this butterfly shape, is all gray matter, and it's basically cell bodies that aren't myelinated, so they don't get or they don't absorb the stain of the Weyert's method. Okay, all clear? Go to the next. Okay, let's go to the next. So now we discussed the types of neurons, and I know you might think this is ridiculous to discuss at this point of the block, but actually histologically you need to know how they look differently. So I want you to visualize them in a diagram, then we're going to take them on to a histology slide, and you want to differentiate between how a multipolar, bipolar, and a pseudo-unipolar neuron looks and where they're located. They ask this question a lot. Where is multipolar neurons located? Where are bipolar neurons located? So you really have to recognize where they're located, how they look, and in histology, how they look in a slide. So first, we have the multipolar neuron. The multipolar neuron, we know how this is how it looks, and this is usually found in motor neurons. On the other hand, we have the bipolar neurons. Then we have the pseudo-unipolar neurons. As we said, the multipolar neuron is found in the anterior gray horn of the spinal cord, meaning it is used for the motor type of neurons. And it's also found in the pyramidal cells of the cerebrum and the cerebellum. So cerebrum and cerebellum pyramidal cells are considered a multipolar neuron. So we can see them in the cerebellum, cerebrum, and the anterior ventral horn of the spinal cord, which indicates that it's kind of mainly for motor function. On the other hand, the bipolar neuron is used for the retina of the eye and olfactory epithelium of the nose. And how I like to think of them, and also the tongue, and how I like to think of them is it's for all the special sensations. So bipolar is for all the special sensations: smell, sight, and taste. Then you have the pseudo-unipolar. The pseudo-unipolar is for the sensation. So it's the dorsal root ganglion of the spinal cord, you can find it, and the trigeminal ganglion of the fifth cranial nerve, which is basically trigeminal. And basically, because the trigeminal is mainly, it has motor, but mainly the thickest part of it is sensory. It's considered a pseudo-unipolar. So again, I want to go back and say multipolar, motor, anterior gray horn, pyramidal cells of cerebellum and cerebrum. Bipolar is for special sensation, retina of the eye, olfactory bulb, and taste, like the special cells for taste. And then you have the pseudo-unipolar, which is for sensation. So you will find them in the posterior or dorsal horn of the spinal cord. And you will also see them in the trigeminal ganglion because it is a heavily sensory neuron. Okay, so now histologically how it looks. This is a multipolar neuron. We said we're going to find them in the pyramidal cells of the cerebrum and the cerebellum. So over here, we have a motor neuron in the anterior gray horn of the spinal cord. This is how it looks, exactly like a pyramid. And you have the cell body over here, dendrites around it, and then the axon over here extending all the way to the bottom. On the other hand, we have Purkinje cells in the cerebellar cortex. So cerebellar cortex has Purkinje cells, and it looks kind of like this hair follicle going out. In the pyramidal cells of the cerebral cortex, you can also see this triangular shape in the cerebral cortex. So just in your brain, basically. So they all kind of look like triangles, and they are multipolar neurons. Then we have bipolar neurons. Bipolar neurons, we said it's for the special stuff. So over here, this is where it looks in the taste buds, okay? And this is where it looks in the olfactory bulb. This is the nerve body over here, or the cell body over here, and then it extends up and down. One line for the axon and one line for the dendrite. So it goes in the middle and then up and down. And the same thing for the olfactory bulb. Then we have the pseudounipolar. The pseudounipolar, as we said, is anterior dorsal gray horn for the sensory. So you can find it in this part of the spinal cord gray matter and in the trigeminal nerve. And what you can interpret from this picture over here is they cut it kind of transversely. So you can see that this is the ganglion cell, this is the nucleus, and this is the nucleolus. And يعني they kind of cut it in the middle, so you can't really see it, the shape of it, like the bipolar and the multipolar. So over here, you have to kind of focus on the top view and how it looks. And yeah. So يعني these are the things you can interpret. It's a ganglion cell with a spherical nucleus. So this is, you see how very circular and spherical it is. It's a large central basophilic nucleolus. You see how dark the nucleolus is inside the nucleus. It's very dark. You have surrounding cytoplasm. The surrounding cytoplasm, and sometimes you can encounter these brownish red lipofuscin granules, which are derived from lysosomes. So you have lysosomes in the cell body, and these these brown red granules are actually indicative of lipofuscin granules from lysosomes. Then you have the satellite cells, which are small cells يعني, that are basically supporting neuronal cells. And these are the things around all these cell bodies around the pseudounipolar. Yeah, I think that's pretty much it for what you need to know about pseudounipolar. I know it's not as clear as bipolar and multipolar, but use it as a distinctive method to be able to know, oh, this is a pseudounipolar. It's located in the anterior horn, the dorsal horn, sensory, and the trigeminal ganglion. So just a reminder of the supporting cells of the nervous system. We have oligodendrocytes, which myelinate the axons in the central nervous system, and squamous cells myelinate the axons in the peripheral nervous system. And just a reminder, oligodendrocytes are able to myelinate more than one axon, while squamous cells are 1-to-1 ratio. So one squamous cell myelinates only one axon. Astrocytes are involved in scarring and forming the blood-brain barrier. Microglia are phagocytic, so they're kind of the immune cell of the CNS, kind of like they act like macrophages. So glial cells are like macrophages. And then you have ependymal cells, which line the cavities of the brains and the subarachnoid space, which creates CSF. And then you have satellite cells, which just regulate the environment. They're kind of the assistants, and they clean around, and yeah. This is an oligodendrocyte where you have one cell body that's able to myelinate more than one axon. As you can see here, it's myelinated 3 axons, while this is a squamous cell with a cell this is a cell body, and one squamous cell is creating it on one axon only. Okay, now I'm going to talk about the cross sections that were mentioned in the lab, and this is kind of an overview of the whole block. So it's very, very detailed, and I go in detail about everything, and I kind of don't skip through it, because instead of spending all the time later in the further labs, I decided to like really give the description here and then kind of reiterate the concept and revise the concept when we revisit them in the next labs. Okay, so bear with me and be interactive يعني كده as much as possible. So يعني let's try to do them together. Someone unmute and tell me, what are these? Number 4. So you're starting from the top. Any guesses what number 4 is? لا تسوي نفسكم ما تعرفوا. You all know this is the lateral ventricle. This is the most basic information in neuro. طيب, you guys don't want to participate? طيب, I'll just keep talking, but you guys are gonna get bored, I'm just saying. طيب, so number 4 is the lateral ventricle. So we have, this is lateral ventricle, this is the lateral ventricle. Over here, we have the superior sagittal sinus. Also over here, the superior sagittal sinus, because remember, the superior sagittal sinus is going all the way like this. It's like a U shape. So it starts here, it goes all the way here. So you're going to see it from the beginning to the end. Because when you cross the section here, you're going to see it from here, and you're going to see it from here. So this is the superior sagittal sinus, also indicating this blue line on the top. And then you have the falx cerebri separating the two hemispheres. This is the falx cerebri. So falx cerebri is number 32 because 35, sorry, because you're dividing the two hemisphere in half. It's just a layer of meninges separating the right hemisphere from the left hemisphere. Obviously, this is all the frontal lobe, and this is all the occipital lobe being the back, and this is being the front. So let's go over it again. 1 is frontal lobe, 2 is occipital lobe, 4 is lateral ventricle, 21 is superior sagittal sinus, and 35 is falx cerebri. All clear? Any questions till now? No questions? Okay. So this is another section that I want you guys to interpret. And basically over here, you can see that the brain is gray matter outside. So gray matter outside and axons inside. So all the axon white matter is inside. And all the myelinated, so they all kind of go inside and then go up to the cell bodies, which are on the top or the outwards. The spinal cord is opposite to that because the white matter is actually, so the gray matter is actually inside, the white matter is actually outside. While here we can see the white matter being the myelinated axons are kind of inside, they branch out going outwards towards the cell bodies, which are gray matter. This is another cross section. I'm telling you, there's a lot of cross section. Bear with me. It's going to be a bit dry content. So that's why it's better for you to interact so that you can really focus with me. But yeah. So over here, we're kind of going below, a bit below, and you can see that the lateral ventricles are kind of going anteriorly. So the number 4 is still the lateral ventricles, but they're much smaller, and they're going down. Okay, and then we have the superior sagittal sinus still in the back, it's visible. Okay, we can still see it's visible. Over here, this crossing thing over here. Okay, and this kind of triangular shape over here and over here. What are these? Can anyone help me? No? Okay. So this is the internal capsule, these two kind of things over here, these flaps actually. Number 28 is the thalamus over here. This is just the thalamus, and this is the thalamus. There are two thalami. Okay, this is the fourth ventricle. I mean the lateral ventricle. Number 27 is not indicated here, so I'm not gonna make up things, but I'm pretty sure it's the basal ganglia, some parts of the basal ganglia, because it's lateral to it. Number 9 is something called the insula, and we're gonna talk now. I'm gonna remind you what the insula is. It's kind of part of the brain that you might have forgotten what it was, but it's okay. We'll discuss it. 33 is connecting between the right hemisphere and the left hemisphere. Do you have any reminder of fibers that connect the right hemisphere and the left hemisphere? Any reminder, any connections? Exactly, it's the corpus callosum. So it's a bunch of fibers connecting the right to the left. So this is corpus callosum. As I said, 4 is the lateral ventricle. 26 is not indicated, and then over here, 29, 30, 31 is internal capsule. This is anterior limb, this is genu of the internal capsule, and this is the posterior limb. Same on the other side. 27 is probably basal ganglia, 9 is the insula, 28 is the two thalami, and 34 is the splenum of the corpus callosum. What is the splenum of the corpus callosum? So like how we talked about in the how the spine what's it called, the sagittal sinus is going all the way up. Same concept is the corpus callosum kind of going in this way. So we have a head, we have a tail, we have a body, and we have splenium, which is kind of the end of the corpus callosum. I'm going to show you a picture of why it's kind of going from here to here, because don't forget, it's kind of going exactly how this is going. It's a U shape from the top. So you'll see it from the anterior and posterior view. Yes, so it's not also called a commissural fiber. It's a type of commissural fiber, because a commissural fiber is things that is connecting the right and the left hemisphere. And we have a lot of types of fiber that connect the right hemisphere to the left hemisphere. But an example of one of these type of commissural fiber is corpus callosum. It's like kind of the main example because it's very big and it connects a lot of information together. So panic, what's the insula? Because when I was doing this presentation, I was like, wait, what's the insula aslan? So as we saw, number 9 is the insula. It's kind of this sulci. The insula is basically a circular sulcus located in the lateral sulcus of Sylvius over here. Okay, it's a functional part of the limbic system. It's a functional part of the limbic system. That's all you have to know, is that the insula is kind of hidden. You have to kind of remove part of the cerebrum to see it. And so you extend these lateral sulci, and then you can see it as the insula, and also known as the island of Rayleigh. And it has functions of like emotional regulation and limbic system stuff. Okay, now we have this is the same diagram. I just wanted to remind you that this is the insula and this is how it looks over here. And it's the same thing as what we saw here in the extension. Okay, what about the external capsule? I forgot everything about the internal capsule. Let's remember it. Let's remember the functions. Let's remember clinical about it, because it's very important. Because internal capsule is tricky and it keeps getting confusing, and I will get any chance to remind you about these functions. So the internal capsule is basically a bunch of fibers. They're a bunch of axons going from one place to another. And it has 3 parts: anterior part, middle part called the genu, and the posterior part. And it looks like it's kind of a wing. So we call it anterior limb, posterior limb, and the genu, which is the middle. The anterior limb carries fibers. What are these fibers? They are the thalamocortical fibers, which go to the frontal lobe. So they're information from the thalamus that want to go to the cortex, or to the front, specifically the frontal lobe of the cortex. So this is the anterior limb. What about the genu, which is this area? This folded area. It has the corticobulbar tract. What's the corticobulbar tract? It's the upper motor neuron of the cranial nerves supplying the head and neck region. Okay, so it's the corticobulbar tract. Basically, the muscles of the head and neck is by this tract. So the fibers pass through this genu. So any damage here, you would lose the fibers. You would have an upper motor lesion of the corticobulbar tract. What would be the presentation? Spastic paralysis of the muscles of the head and neck. Okay. Then we have the posterior limb. The posterior limb is kind of the longest one. It's very clinically relevant, so you kind of have to know the information about it. It carries the corticospinal tract. What's the corticospinal tract? The corticospinal tract is basically the motor tract of the rest of our body. We talked that the motor tract of our face and neck is the corticobulbar. Now the corticospinal is in the posterior limb. It regulates the motor of the rest of the body, moving our hands, moving our legs, and all of these things. So losing the posterior limbs, meaning you will lose the ability to move in any part of your body except the face and the neck. Okay. This is the corticospinal tract, so things pass through it. But it's kind of arranged in that this area that's close to the genu, the posterior limb that's close to the genu over here, kind of do the muscles from the upper limb more. And then as you go more posteriorly, so this area, you would have the lower limb. So if you have an injury here, you would kind of have paralysis of the lower limb, while if you have an injury very near to the genu, you would have probably loss of motor function of your upper limb. Okay, so the closer you are to the genu, the more upper you are. The more posterior, or as you go down the posterior limb of the internal capsule, the more you're in the lower limb. The other that we talked about the corticospinal tract, it also carries another important tract, which is the thalamocortical fibers. So also it's sending information from the thalamus to the cortex. And it sends them to the postcentral gyrus, okay, of the parietal lobe. So again, let's repeat. Anterior limb is responsible of thalamocortical fibers. Genu is for corticobulbar tract. Posterior limb is corticospinal tract and thalamocortical fibers. How are they organized? Near to the genu is upper limb, near the lower in the posterior limb, or more posteriorly, is the lower limb. We also have two more parts to the internal capsule called the retrolentiform and the sublentiform areas. And basically, they're a bunch of fibers that are crossing from the geniculate body to another part of the body. So we have the medial geniculate body fibers going from the medial geniculate body. The fibers are going to the auditory radiation in the temporal gyrus, because we know the temporal lobe is here and it's near the ears. So obviously you want the fibers to go towards the ear. So you're going from the medial geniculate body of the thalamus to the auditory radiation. This is part of the internal capsule called the retrolentiform. So what is it doing? Taking information from the this is the sublentiform. It's taking information from the medial geniculate body and sending it to your ears, which is the temporal auditory cortex. It's also known as Brodmann areas 41 and 42. Then we have the lentiform part, the retrolentiform part. This is for the lateral geniculate body. Lateral geniculate body, L for light. So it's for vision. M for music. So for music. So medial geniculate body is for auditory radiation. Lateral geniculate body, L for light, is vision. So you're studying visual radiation to the occipital lobe, which is responsible of your vision. Okay, so retrolentiform is lateral geniculate optic radiation to occipital lobe, or Brodmann area 17. Sublentiform area is auditory radiation from the medial geniculate body to the temporal auditory cortex. Okay, but if you're confused why we call it retro and why we call it sub, it's just the location of the geniculate body. One is under the lentiform and the other is behind the lentiform. So the letter L, the lateral geniculate body, is behind the lentiform. The sublentiform is basically medial geniculate body under the lentiform nucleus, which is part of the basal ganglia. Excuse me. Just gonna drink water. Okay, so this is kind of a region of the internal capsule. And I told you guys, I'm gonna go in detail to everything. So now we go back to 29, 30, and 31. If you have injury of 29, you'd know what's broken. If you have an injury of 30, you'd know what's broken. If you have injury of 31, you'd know what's broken. If you have injury of this area, which is actually, what is it? Optic radiation and auditory radiation, you'd know what's broken. Okay, so this is the teaching style. And I'm gonna do clinical. So what's clinically relevant is the genu and the posterior limb. This is what you kind of have to know the blood supply to. So the genu is, as we said, corticobulbar tract. So it's up the motor neuron of supplying the muscles of the head and neck. Okay, it's supplied by the anterior choroidal artery and the lateral lenticulostriate arteries. And this lenticulostriate artery is part of or a branch of the middle cerebral artery. So both of them. So both of them are middle cerebral artery middle cerebral artery brings up anterior choroidal and lateral lenticulostriate, and they both supply the genu. Okay. If you have a stroke, a lacunar stroke in this area where you're damaging the genu, over here, I'm gonna show you. Do you see this darkness, this dark L shape? If you damage the middle, or over here, you damage the middle over here. This is damaging the genu. If you damage the top dark part, it's the anterior limb. And if you damage the bottom part, it's the posterior limb. Okay? So, if you have a stroke of that, we said that it's for the corticobulbar tract. What did we say the corticobulbar tract is responsible of? What type of muscles or what type of motor response? I need someone to answer me. Yalla, I'm gonna wait now. More energy, more energy. Nothing. ما تعرفوا ايش يسوي الجنيو؟ وين الحماس يا جماعة؟ وين الحماس؟ طيب. The
What?
genu, yeah, go ahead.
I said corticobulbar.
Yes,
Bulbar.
corticobulbar. And the corticobulbar is responsible for the muscles of the head and neck. So what's one of the muscles? The uvula and the tongue. Okay. So that's kind of the main symptom that you will focus on. And when you lose the fibers in the internal capsule, it's an upper motor neuron lesion because it's not in the face itself. You're going from the top, the upper motor neuron. So what would it be? It's going to be spastic type of paralysis, right? So you have spastic type of paralysis of the corticobulbar tract, and you will have facial paralysis, okay, of the facial nerve, the facial nerve over here. It's going to be the lower part of the face only because the forehead has dual supply. So this area over here is not going to be paralyzed because it's being supplied by both sides, by the right and left nerve. Okay, but the lower part only is supplied by the contralateral nerve. So for example, this is my right. This is my right side. Okay, so it's supplied by the left nerve. And this left side supplied by the right nerve. My forehead is supplied by both the right and left. So it won't have paralysis. Even if the right is broken, the left will supply it. The lower is supplied by only one nerve. So if I lose my left, my right will be drooping. So I won't have muscles here. So I'd have left-sided. So in this case, where the injury over here is on the right side of the patient, would be on the right side, it would be left-sided lower facial paralysis. Okay, with sparing of the forehead because of dual blood supply. Sorry, not dual nerve supply, dual blood supply to the nerves. So then you won't have a stroke to it. So the nerves will be alive, basically. And the you will have lower facial paralysis, and then for the uvula and the tongue, you will have a special presentation. What is it? As we said, this is spastic type of paralysis. Okay, so if it's spastic, okay, the uvula will deviate towards the lesion, while the tongue will deviate away from the lesion. Why? الحين you're gonna get confused. Why? Think of it. Let's say I have a lesion on the يعني كذا, my left side is broken. My left side is facial paralysis. Okay, which nerve is broken? My right nerve. Okay, my right nerve. Okay, so my uvula is spastic. That the muscle will stretch, will be broken, and it will go towards the right side, لأن ما في supply to the left side, so it will تنشد towards what's working and towards the right side, which is working. So then the left side is broke. I confused you guys, I know. يعني everyone has to understand it in their own way. فأنا ما بتفلسف أعلمكم طريقتي. I have like a certain way I can imagine it. So I'm not going to explain it again because I feel like I just confused you guys. Uvula, lesion towards upper motor neuron lesion, tongue towards away from the lesion. Yeah, towards the lesion. So the uvula towards the lesion, tongue away from the lesion. And it's kind of all about, like, if you really think of how the muscle is being stretched when something is working, something's not working, it's going to make sense. I just can't, I don't know how to explain it, so I don't want to be responsible of confusing you. But this is the presentation of the genu. طيب. أما clinical feature of the posterior limb, as we said, the posterior limb is responsible of the corticospinal tract. Corticospinal tract is responsible for the muscle of the rest of the body, so all your muscles of the body. So if it's broken, you will have paralysis of all the muscles on the side of the opposite side of the lesion. So if you have, for example, right-sided paralysis, it means you have left-side stroke. Obviously, it's common sense, contralateral. Okay, what is the posterior limb supplied by? It's supplied by the exact arteries of the genu. It's supplied by the anterior choroidal artery and the lateral lenticulostriate artery, which are both branches of the medial cerebral artery. The lacunar stroke of the posterior limb would show contralateral hemiplegia and contralateral hemianesthesia. So you won't have sensation, and you won't have what's it called? You won't have يعني the movement of you have paralysis, spastic paralysis. And the reason why you have the hemianesthesia or lose the sensation, although we focus on the corticospinal tract and not the other tracts that are responsible for sensations, because the thalamocortical fibers that are coming from the ventral posterior nucleus of the thalamus are sensory, and they're actually over here we said they're going to the postcentral gyrus of the parietal lobe, which is a sensory gyrus. So if you're losing the fibers that are going to the sensory, it will never reach the sensory gyrus or the postcentral gyrus. So that's why you will have loss of sensation, not necessarily because the spinothalamic was lost or the dorsal column was lost, no, but because it didn't even reach to the postcentral gyrus, which is the sensory gyrus of the brain. So that's why you lose the sensation due to the thalamocortical fibers coming from the ventral posterior nucleus of the thalamus. طيب, yes, basically that's all. And because they have similar supply, you can have a case where the genu and the posterior limb are affected together because they have the similar supply. But you're just gonna have to see the symptoms in order to conclude what type of يعني occlusion or stroke that you have. Okay? I know this was kind of يعني طيب, what about clinical scenario for losing the medial geniculate or like the optic radiation or auditory radiation losing it. Obviously, you would have contralateral homonymous I can't say it, but you guys get it. Basically, you lose this half your eyesight, and or you will have loss of bilateral hearing loss, slight loss of bilateral hearing loss, if you lose the auditory radiation. But what supplies them? For the optic radiation, which we said optic for eye for visual and L for light. So this is the lateral geniculate body. If you lose that, then it's supplied by the posterior cerebral artery, while the auditory radiation, which we said M for music, medial geniculate body, it is supplied by the anterior choroidal artery. So if you lose the anterior choroidal artery, you would lose the medial geniculate body, and you would lose the auditory radiation, and you will have, obviously it has to be bilateral, and you would have bilateral hearing loss. Okay. So again, we talked about all these things. So we talked about the lateral ventricle, we talked about the superior sagittal sinus, we talked about the insula, we talked about the anterior genu and posterior limb of the internal capsule. Now I'm going to remind you of the splenium of the corpus callosum. The splenium of the corpus callosum, I said the corpus callosum is similar to the concept of the sagittal sinus, superior sagittal sinus going all the way up. This is also a picture of the corpus callosum going all the way around, and you have the rostrum, the genu, the body, and the splenium. And it was talking, and this specific protrusion in the splenium is what you see over here in 34, and what you see in 33 is probably the genu, or probably the genu. So you have kind of this type of cut, or the rostrum. It's basically just parts of the corpus callosum. Okay. Now this is another section. This is basically so let's orient ourselves over here. This is the occipital lobe, okay? And number 10 is this, that's when you start to see the cerebellum, okay? You're starting to see the cerebellar hemisphere. And what's in front of the cerebellum? You can still see the brainstem. So this is kind of, number 6 is kind of indicating the midbrain level. And in the midbrain, which is number 36, there's this darkness. And we all know that in the midbrain of the brainstem, we have the substantia nigra, which is part of the basal ganglia. Substantia nigra is significant in Parkinson's disease because they have this, instead of this dark brown blackish color, they have light brown, meaning they have less dopaminergic neurons. So this dark color, this black color, indicates dopaminergic neurons being available. If it's very light and faint and there's no actual color here, then you mean you have Parkinson's disease because you don't have enough dopamine neurons in the substantia nigra in the midbrain. So again, this is all the midbrain. And then we have number 40. 40 is the red nucleus, and it's just available in the, it's just the nucleus that is in the midbrain, and it is responsible of the rubrospinal tract, and the rubrospinal tract facilitates the flexors, okay? So again, and then number 1 is the frontal lobe, okay? This is the frontal lobe number 1. دقيقة presentation علق. Okay, again, I'm going to revise. This is the occipital lobe. This is the start of the cerebellar hemisphere. Okay, this is the brain stem. Over here, we're in the level of the midbrain. How do we know? We can see the substantia nigra. These dark dopaminergic neurons. 6, as we said, indicates that we're in the midbrain, and number 40 is the red nucleus in the midbrain, responsible of the rubrospinal tract, which is responsible of the facilitating the flexors of the body. Okay. And this is the rubrospinal tract. We said this is the red nucleus in the midbrain, and it's responsible, as we said, for the flexors. So you're going from the spinal cord to the, sorry, I think it's a descending track. Yeah, I think it's a descending track. So you're going from the brain to the red nucleus, from the red nucleus contralaterally to the spinal cord, and eventually to the flexors of the muscles. Okay. Now we have another section. Number 3. Okay, so over here you can see clearly number 10. These are the hemispheres of the cerebellum. Okay. Number 7, this is still the brainstem, and we can see the pons. Okay. Number 3, these are just part of the brain. So you're kind of seeing the temporal part of the brain. You can see the eyes here, you can see the nose and the olfactory area, the sinuses, okay? What are these two muscles on the side? You're kind of at the level of the jaw, kind of. So this is called the temporalis muscle. The temporalis muscle helps in mastication. So it's a muscle of mastication, and it is supplied by the mandibular nerve. So this is called the temporalis muscle, okay? Over here, we said this is the cerebellar hemisphere. And we said we're at the level of the pons. And in front of the pons, we know that there is a groove on the pons called the basilar groove. And the basilar groove carries the basilar artery. So number 14 is the basilar artery. And we see over here, we know that this is the superior sagittal sinus initially, this area. But then it divides when you go down at the level of the jaw level, you're talking jaw level, it's actually separated and became the transverse sinus. So this is the transverse sinus, and this is where the transverse sinus is. And this is the transverse sinus, kind of like from superior sagittal sinus, it divided into longitudinally into the transverse sinus. In the front over here, in the temporal lobe, or this area over here, you can see the yellow one, which is number 11. It's an artery. Yellow is always artery in these cases, and it's the internal carotid artery. On the other hand, what's opposite to it is the internal jugular vein. So this is the vein, and this is the artery. But this long line over here in the pons, this is an important line. It's a nerve. What's the nerve that comes out كده in the middle of the pons? My best guess is this is the trigeminal nerve, because it's kind of coming from the vestibulocochlear. صح صح. It is the vestibulocochlear. I'm not 100% sure of the level, but it could be that. Vestibulocochlear. I thought trigeminal because it's in the middle. But no, you're right. It's vestibulocochlear. And in next slide, Aslan, we're going to indicate what this line is. I just don't want to say it for myself. It's going to be in the slides, which nerve this is. But this is definitely a nerve going out of the pons. So I'm going to revise again. 19 is superior sagittal, still superior sagittal sinus. 15 is transverse sinus. 10 is cerebellar hemisphere. 7 is pons. In front of the pons, we have the basilar groove creating basilar artery. And then we have the vein over here, the internal jugular vein. Then you have the internal carotid artery. And then what does the internal carotid artery give out, guys? Gives out the middle meningeal artery. And then number 5, we said this is the temporalis muscle. We said this is the temporal lobe of the brain. And I think that's all that we have to know in this section. Okay. Oh, and just something that you should know is that over here, we have this kind of dura mater that separates between the right hemisphere and the left hemisphere of the cerebellar hemisphere. So even the cerebellum has kind of a dura mater that separates both lobes, called the falx cerebelli. Okay, so just كده context. We said إن this is the superior sagittal sinus. كده focus on the red laser, okay? Superior sagittal sinus, and we reached to a point where we have the confluence of sinuses. This might, depending on the level, this could be the confluence of sinus. This could be the sagittal sinus. I can't identify it. But what you have to know is that when it separates to right and left, it's for sure the transverse sinus. And it's because you can see it on the right and the left over here. Okay, and the transverse sinus, when it goes down, it goes down to become the sigmoid, and then to the internal jugular vein. And where's the internal? So it goes all the way here. It goes down and it becomes in the internal jugular vein here, number 12. This drains into this. In terms of the internal carotid artery, we said the internal carotid artery comes out. This is the internal carotid artery, and it forms the middle meningeal artery. This is the internal carotid artery. It creates the middle meningeal artery.
Middle cerebral.
Middle cerebral or middle cerebral artery, sorry. Yeah. So it creates the middle cerebral artery. Yeah, it's okay that I didn't forget anything. Basilar. Oh, and then something else we said 14 over here is the basilar artery, number 7. So this is the basilar artery, and you can see this is the pons, these two circular things, and in the middle we have the basilar groove, and this is the basilar artery, and this is also the basilar artery. These are the pons, and this is the basilar artery. Okay. Yeah, so next. Then we have, this is kind of the concept of the dura that I mentioned. I said that over here we separate the cerebellum with the dura mater called the falx cerebelli, and this is where it is. It's kind of this dark area over here. This is falx cerebri, which separates the two hemispheres. This is falx cerebelli, separating the two hemispheres of the cerebellum. And then we have the tentorium cerebelli, separating the cerebellum from the cerebral hemisphere. This would be the cranium part where the occipital lobe, this is the area where the cerebellum would lie under the tentorium cerebelli. And yeah, that's all. And this hole is for the brainstem to go in. This hole in the cerebelli, the tentorium. Okay, this is another one, another section that you have to know. As we know, these number 10 is the cerebral hemisphere. Okay, and in the cerebellar hemisphere, and in the cerebellar hemisphere, what's the middle part that kind of divides the two hemispheres divided? We know the meninges, but in terms of part of the actual cerebellum, it's the vermis, what's in the middle, right? The number 16 is actually the vermis of the cerebellum. Number 41 is the dentate nucleus, and I don't know if you remember, the dentate nucleus is one of the nuclei of the cerebellum. It's actually one of the nuclei of the cerebellum, and it looks like a C shape. So I don't know if you noticed, this yellowish thing is kind of a C shape. So you can see it here. This is a C shape, which is the dentate nucleus of the cerebellum. Okay, and the blue line is the trigeminal nerve. So actually, I think I was, when I said the option before, I think I was right about it, and this is the trigeminal, because you can see أصلا how thick the nerve is. Oh, أصلا, yeah, it's coming out of the number 7, which is the pons. This is the temporal lobe, and number 11 is the internal carotid artery, which we said. We said this, and this blue thing number here over 23 is the cavernous sinus. The cavernous sinus is where kind of a lot of nerves combine together. But the issue with this sinus, where a lot of veins drain into it, is that it's close to the eyes, and it's close to the eyes, and I think I'm not sure, we're gonna say it in the next, wait, دقيقة. Yeah, so any infection or thrombosis in the eye or the face has a chance in spreading of in the cavernous sinus, because it actually collects. So over here, this is the cavernous sinus, what we see over here in 23. And it collects, drains the ophthalmic vein from the eye and the facial vein from the face. So any infection that goes here can spread in the cavernous virus. And the cavernous virus actually eventually drains together in the internal jugular vein. So you're kind of creating a whole problem. And that's why cavernous sinus is very significant clinically. That you have to know where it is and why it's clinically relevant, because it's any infection or thrombosis that happens in ophthalmic vein or facial nerve can spread in the cavernous sinus, which can spread all over in the venous system in the brain. Okay. And we said this is the dentate nucleus. This is basically what I'm talking about, the C shape over here. And this is exactly how it looks over here in 41, and this green color over here. So I think it's very interesting to compare the two. We said the middle over here is the vermis, and this is number 16 is also the vermis. And these are the two hemispheres. And this is the trigeminal nerve. This is still the basilar, number 14. 11, internal carotid artery, cavernous sinus, temporal lobe, and then this is the temporalis muscle. All clear till now? Any questions? Okay. We said, so okay, here we have to know that at this point, we're at the level of the medulla. Okay, now we're at the level of the medulla, so we're a bit bottom. So خلاص the basal artery is, we're going down. So basal artery is actually made up of two things. What are the two things? Two vertebral arteries. So over here, we have this is the vertebral artery. We have vertebral artery that create the basal artery that moves on the pons. So at the level of the medulla here, it's still the vertebral artery. So actually number 38 is the vertebral artery. 8 is the medulla. And then over here, when you see in the middle, in the groove, you can see that this is the pons and this is the basilar artery. So you can see that in the medulla level, it's vertebral artery. And then when it goes all the way to the pons and becomes in the basilar groove, it combined and became the basilar artery, and eventually goes up to become other stuff. Clear? Next slide. Okay. So let me see how many slides to give you guys a break. Okay, we'll take a break after we finish these two sections. Okay, we'll take a 10 minutes break because we have a long way and I need you guys to continue focusing. So number 18 is okay. Now we are going up again. And we're at the same level where we saw the cavernous sinus. We said 11 is the internal jugular vein, and internal carotid artery. And this is also the internal carotid artery. In the middle over here, we have the pituitary gland sitting on the sella turcica bone. So this is actually where the pituitary gland is located. Okay. We know all these things. We discussed them a lot of times. Number 20 over here is the sigmoid sinus that we talked about here. The sigmoid sinus, where we're going down this way. So actually, when we're going this long, number 24, it's the sigmoid sinus at this point. And also number 20, where it's a bit thinner instead of this, it's the inferior petrosal vein. And over here, it's not so over here the inferior petrosal it actually takes things from here and it drains it to the sigmoid. So actually they meet up in the middle. So if you really think about it, the flow is going from here and then draining to the sigmoid vein at 24. Okay, so you're going from the petrosal sinus, inferior petrosal sinus, and draining at the sigmoid sinus. And we said this is the pituitary. The rest we said 23 is cavernous sinus, 11 is internal carotid artery. And this 41, we can see the caudate nucleus, cerebellar hemispheres, 7 is pons, and 17 and 16 is 16 is vermis. 17 is probably fourth ventricle, I'm not sure though. Okay, so this is the inferior petrosal artery. We just talked about the superior inferior petrosal artery draining into the sigmoid artery. I mean vein. Okay, now 21. we said is superior sagittal sinus. It's not anything hard. And over here, we said it's the corpus callosum. We already talked about it before in other slides. Okay, now we're going to talk about the types of fiber. Just a basic reminder, we said commissural fibers connect both lobes. So an example would be corpus callosum. We're going from the right to the left or left to the right. And then we have short association fiber from one cell tied to another next to each other, separated by a gyrus. And then we have long association, which is from, like, for example, from here all the way to here. That would be a long association fiber. And then we have a projection fiber, which is all the tracks we learned. So things that are going up from the brain to the spinal cord, vice versa, spinal cord to the brain. And we'll take a good 10-minute break على ما نكمل. يعطيكم العافية. Don't worry, questions are coming. This is all just organization of the nervous system, but trust me, it gets much faster when we finish this part. It's very heavy, I understand, and I'm sorry about it. Yalla, you could stop recording, Muhammad, and we'll be back at 11. Let's do a 7-minute break. Okay. فالحين we're going to talk about the bones, basically kind of like the insertions of the nerves, the types of bones, and all these كده things that you wouldn't study, but then they come in the exam. That's كده my personal experience. But basically, let me get my laser thing. There's this. Yes, okay. One laser. Okay, so identify the bones. We know this. Frontal, occipital, parietal. The butterfly-like yellow thing is called the sphenoid bone, and the red one is called the temporal bone, obviously. The purple one over here, the small one, is called the ethmoidal bone. That's all. So the frontal bone, what you have to know is that, oh, and just كده context, first part over here is the anterior fossa, middle fossa, posterior fossa. Okay. Over here we have in the frontal lobe, what you have to know is that it has the orbital plate. So over here you have your eyes. This is where your eyes come out. Okay. So يعني this is orbital plate that holds the eye. This is kind of we won't talk about the fissures here because I think it's much easier for you to see it as an overview. This is just a model that I want to show you from lab, where you have the falx cerebri, the superior sagittal sagittal sinus, and then the tentorium cerebelli that separates the cerebellum, and then cerebri that separates the cerebellum from the cerebrum. Then we have the ethmoidal bone. The ethmoidal bone is where the crista galli is, which is this purple thing. And just so you know, in the crista galli, which is the protrusion in the ethmoid bone, this kind of like plate, is where the dura mater, like it attaches. The falx cerebri that separates the two hemispheres attaches there. And also it has olfactory nerve bundles and foramen to pass like smell and stuff like that. Then we have the sphenoid bone, which has two wings. We have a lesser wing and we have a greater wing. We have a body. Just what's clinically important is that it has the sella turcica, which is kind of a chair for the pituitary gland. It holds the pituitary gland. Then we have the temporal bone, which is the red part. It has a flat part and a protruding part. The flat part is called the squamous part, and the protruding part is called the petrous part. And you can see it here in the model. This is the squamous part, and this is the petrous part. Then we have the occipital, which is the orange. What you have to know is that the internal occipital protrudence, which is over here, is where the falx cerebri attaches posteriorly. So we said anteriorly, it attaches to the ethmoid bone over here, and posteriorly, it attaches to the occipital bone in the internal occipital protrusion. And yeah, and it has the internal cranial occiput, and then it has the foramen magnum over here for the spinal cord and the brainstem to come in. As I said, this is the anterior cranial fossa, middle cranial fossa, and posterior cranial fossa. I said that over here we have the frontal bone. This weird small thing is the ethmoidal bone for the olfactory bulb and so on. This is, we said, the butterfly, which is the sphenoidal bone and has the sella turcica over here where the pituitary gland can sit. Then we have the occipital bone, and in the back, we have the internal occipital protrusion over here, which connects the bulk cerebri from here all the way to the cryptiform plate of the ethmoid bone. Okay, or was it the cristagalli, sorry, not the cryptiform plate. The cryptiform plate, olfactory nerves. The cristagalla is the falx cerebri. So over here, when you see the cristagalla, which is on the top, it's where the attachment occurs. The sides where there's holes, which is the cryptiform plate, is the olfactory bulb area. Okay, all clear till now for these like bone features? Any questions? Okay. الحين we're going to talk about the insertions of the nerves. Okay, so first we have the cryptiform plate. It's not relevant for us because the first cranial nerve comes from out of it. But now if we go to the middle cranial fossa and the posterior cranial fossa, it's where we need to know stuff. Okay, I want this picture, it's better for me. So the optic nerve comes out the optic canal over here on the top. And it would be equivalent to this one. And equivalent to this one. Okay, this is the optic canal, where the second cranial nerve comes out. Okay, then we have the superior orbital fissure, this long, slender thing. Okay, superior orbital fissure has 3 nerves and a half that come out of it. 3rd, 4th, 5th, which is oculomotor, trochlear, ophthalmic, and abducent nerve. And the ophthalmic nerve is a branch of the trigeminal nerve. Okay, again, oculomotor, trochlear, abducent, and a branch of the trigeminal called the ophthalmic branch. And this is where it is this long, slender thing. And I'm pretty sure it's a long, slender thing here, but it's not showing. It's also here, superior orbital fissure, optic canal on the top, superior orbital fissure here. Okay, then we have 1, 2, 3 holes. So we have superior orbital fissure and then foramen rotundum, foramen ovale, foramen spinosum. You have 1, 1, 1. The first one, which is foramen rotundum, carries the second branch of the trigeminal nerve. What's the second branch of the trigeminal nerve? The maxillary branch of the trigeminal nerve. And the third one, which is foramen ovale, carries the third branch of the trigeminal nerve, which is the mandibular branch of the trigeminal. Okay, so we can go here and we say again. First one, optic canal, second cranial nerve. Third one, superior orbital fissure, carries 3 nerves, cranial nerve 3, 4, and 6, plus 1 branch of the trigeminal, which is the ophthalmic branch. Then we have foramen rotundum, which carries the second branch of the trigeminal, which is the maxillary branch. And then the last over here, the foramen ovale, which looks like an oval, carries the third branch of the trigeminal, which is the mandibular branch of the trigeminal. Then we have the foramen spinosum. The foramen spinosum is right next to the ovale, and it carries the middle meningeal artery. Middle meningeal artery. Then we have internal acoustic meatus. From the name, what would it carry? It would carry the vestibulocochlear nerve because it's for hearing, and facial nerve. Then we have the third, and then we have, where is this one? And then we have the jugular foramen, which carries out cranial nerve number 9, 10, 11, which is glossopharyngeal, vagus, and accessory. Okay. And then jugular, and then I think jugular vein. Yeah, and the jugular vein. Then the one over here is the hypoglossal canal, which is for cranial nerve number 12, which is hypoglossal nerve. Then you have foramen magnum for the brainstem, the spinal root of cranial nerve number 11, and vertebral artery. I don't know if you guys remember, but 11 had a cranial and a spinal branch. And the spinal branch comes out of the foramen magnum. And we have vertebral arteries. Okay, we're gonna revise this again in this diagram. Over here we have the optic canal, which gives out the optic nerve and ophthalmic artery. Then over here we have the superior orbital fissure, which is the long one, and it creates three nerves: oculomotor, trochlear, and abducens. We're all for the eye. And superior ophthalmic division of trigeminal. Then we have foramen rotundum. Foramen rotundum is for the second branch of the trigeminal, which is mandibular. Then, maxillary. And then foramen ovale, which looks like an oval or a P shape, is for the mandibular branch of the trigeminal. And then foramen spinosum, which is this tiny part, is for the middle meningeal artery. And then foramen lacerum is for, and then over here, this I didn't mention it. This over here, foramen lacerum and carotid canal is for the internal carotid artery. Then we have the internal acoustic meatus, which said for the what? Things for the hearing, for the vestibulocochlear and facial, because facial also is involved in hearing, if you really want to think about it like that. And then the hypoglossal canal over here is for the hypoglossal nerve, and the foramen magnum is for the brainstem, the meninges, and the vertebral arteries and the spinal root of the accessory nerve or 11th cranial nerve. Okay? So this is kind of what you have to know. The septum pellucidum is what divides the two lateral ventricles from each other. This is the septum pellucidum. I think this is enough. Okay. We talked about this kind of, if you want a reminder. Oculomotor, and then we have the optic first, oculomotor, trochlear, trigeminal, abducent, vestibulocochlear, hypoglossal, accessory, vagus, plus pharyngeal. We're gonna go over them more in depth with their functions. But does anyone want to help me test ourselves from each foramen? Which nerve comes out? Optic nerve, second cranial nerve comes out which foramen?
Was it the optic canal?
Yes. And then the second, there is one, the next one, what is the foramen and what comes out of it?
The superior orbital fissure. It carries the oculomotor, trochlear, abducens, which is 3, 4, and 6, and the ophthalmic division of the trigeminal, which is V1.
Amazing, amazing. And then what comes after it? Which foramen?
Foramen rotundum.
Yes.
And it carries the second division of the trigeminal, V2, which is the maxillary branch.
Okay, and then the third one?
Ovale.
Ali.
Carries the third division of the trigeminal, the mandibular.
Yeah.
And
then
then
after,
spinosum,
yeah.
the middle meningeal artery, and كان internal acoustic meatus, internal acoustic meatus facial nerve 7th, and the vestibulocochlear nerve the 8th.
Yeah.
بعدين في the jugular foramen, which carries the jugular vein, internal jugular vein, and the cranial nerves 9, 10, and 11.
صح. Okay. And then the last one, which is hypoglossal canal for the hypoglossal or 12th cranial nerve. Perfect.
Yeah.
Thank you so much. Thank you so much.
في foramen lacerum and something else.
Yeah.
Carotid
طيب.
canal.
Carotid canal for the carotid artery, internal carotid artery. And foramen lacerum for, do you remember? It's for the foramen lacerum for the foramen lacerum and carotid artery for internal carotid artery. Okay. And then the foramen magnum, which is for the brainstem and so on. Okay, good job. So this is kind of like a random insertion I put because they tend to ask يعني about the embryological يعني derivative of anything they ask about. So if they ask even about the basal ganglia, they'll be like, oh, where does it come from? So just this is a brief reminder. We have procephalon, mesocephalon, and rhombocephalon. Procephalon divides ventrally into telecephalon and dicephalon. Telecephalon gives the cerebral cortex, basal ganglia, hippocampus, olfactory bulb, and basal brain. What's important is to know what ventricle as well. So telecephalon creates the lateral ventricle. Dicephalon creates the thalamus and all the thalami, hypothalamus, epithalamus, all the thalami بالحياة, dicephalon. And third ventricle. Why? Because thalamus T and third is T as well. Okay, all the T's are together, dicephalon. And then mesencephalon is for the midbrain and cerebral aqueduct. Okay, this is important. Cerebral aqueduct for mesencephalon. Then rhombocephalon for metanecephalon and myelocephalon. Metanecephalon is for cerebellum and pons and the fourth ventricle. Myeloencephalon is for the medulla and the fourth ventricle. So if they ask you the fourth ventricle comes from where, you can just say the rhombocephalon. Then the spinal cord, obviously associated with the central canal. Okay. Okay. Now let's do questions. A bunch of questions. We're gonna do a lot of questions. الحين أنا من جد ما بجاوب عنكم. كلكم لازم تجاوب. يلا. What is the type of neuron pointed at? And what morphological changes are seen in the neural cells death? يلا يا
Is it
جماعة
random?
يلا قدها. It's ايش؟ Yes. So Ahmed answered multipolar. Sorry, I didn't hear you. But it is
I said,
multipolar. ما سمعت. Sorry. It is multipolar neuron. So it is a multipolar neuron. Oh my gosh, I just realized it's written. I'm sorry. But what's the morphological changes seen in neural cells? This is kind of a tricky question because we didn't mention it. But I will refresh your memory because it's a pathology question. But it comes in OSPE.
Red neurons?
Yes, exactly. Red neurons. Good job. So multipolar neurons and red neurons. طيب. As I said, as we said, this is kind of reminder, this is how a multipolar neuron looks like. It looks like a pyramid. These also look like pyramid cells over here. Not as clear, بس they do the job. And we said they're located in the anterior horn of the spinal cord, cerebellar cortex, and cerebral cortex. And about the red neuron, it's after 12 to 24 hours of irreversible hypoxic or ischemic death, you will have red neurons which shrink the cell body, have pycnosis of the nucleus, disappearance of the nucleolus, loss of nuclear substance, and eosinophilia of the cytoplasm. You can see this is a normal pyramidal cell, and this is a red neuron after 12 to 24 hours. At which area does the ophthalmic pass through? Optic fibers قصدي. Any guesses? Okay, it's okay. So the answer is 37 قصدي. So the answer is 37 because remember we said the optic fibers is coming from the medullary mammillary body, I think. Lateral geniculate body. From the lateral geniculate body for the optic fibers for the visual like pathway. 37 is where the retrolentiform fibers are over here. And 33 also it doesn't make sense because 33 is the posterior limb and optic fibers don't go through the posterior limb. 35 is the anterior limb and it doesn't make sense for it to go there either. So 37 is the answer, and this is a reminder in 37 over here. It says retrolentiform fibers, and we said retrolentiform fibers are optic radiation axons from the lateral geniculate body running to the occipital visual cortex, which is over here. Okay. The answers keep coming up, sorry. Which the vestibulocochlear nerve passes through which canal? We said vestibulocochlear, which canal? Or which other nerves pass through it as well? واسم الكنال, please. Okay. So it's actually B because the nerve that passes through it is also the facial nerve. And we said this is called the internal acoustic meatus, number 6. D is, I'm pretty sure the D is the foramen ovale, which has the second branch of the trigeminal next to it. And then and C is probably the lacerum, which is internal carotid artery comes out of it. A is probably jugular, which means it's the jugular vein comes out of it, and cranial nerve 9, 10, and 11. So this is a kind of a reminder in internal acoustic meatus over here, and same as B is where the facial nerve and vestibulocochlear nerve comes out. And this is kind of a reminder, we just mentioned it, so I'm not going to go over it again. Okay, what about this question? Anyone want to answer? Blood supply of C. And what is C? Okay.
C is the posterior limb of internal.
Yes, perfect. And what is the blood supply?
Lenticular striated artery.
Yeah, and or the anterior choroidal artery or both of them. So this is the blood supply, lateral lenticulostriate artery and the anterior choroidal artery. For the identifying, this is the posterior limb, this is the genu, and this is the anterior limb. So just a reminder of the clinical, posterior is responsible of the corticospinal tract, which is the upper motor neuron of the anterior gray horn cells. And in addition to the thalamocortical fibers coming from the ventroposterior nucleus, meaning they're responsible of going to the postsensory gyres, which is sensory. طيب. It's supplied by the anterior choroidal artery and lateral lenticular striate artery. Lacunar stroke to the posterior limb would cause contralateral hemiplegia of the due to loss of corticospinal tract, contralateral hemianesthesia due to loss of thalamocortical tract that are going to the postcentral gyrus or the sensory gyrus of the brain. Okay. طيب, which of the following cells are responsible for motor activity? So I want you to tell me, what are you looking for? Which cell are you looking for? And which one it is in the picture?
Okay, so we're looking for motor neurons.
What
From the
type of neurons are motor neurons in terms of like shape?
multipolar,
Multipolar.
and I think B. The answer would be B.
Yes, perfect.
Sorry, the picture on the right is the layers of the cerebellar cortex. So we'll choose that on the left, and B is the answer.
Yeah, perfect. Good job. So yeah, you're completely right. So the primary motor function of the body is by the pyramidal cells, which are done by in the cerebrum. And this is kind of a reminder. And you're looking, and who هنا you found in the cerebellum are Purkinje cells. I wanted to show you that these are the Purkinje cells. Okay. Oh, I didn't put a transition to this. Okay, so basically, if you have spastic paralysis of the upper and lower limb, it was just asking, where to point at it. But the answer is here because it's the posterior limb. Because remember, the posterior limb is responsible of the corticospinal tract. Okay. A patient presents with dropped face on the right, and the injured nerve leaves the skull from which of the following foramens? Any guesses?
Is it the facial nerve?
Yeah. Facial nerves comes out of
Yeah, internal acoustic meatus.
B. Good job, B. Yeah. Internal acoustic meatus. It's again a reminder. This is the internal acoustic meatus over here. It comes out as facial and vestibulocochlear. Okay. A 60-year-old man walks into a clinic with stooped posture and rigid facial expressions. He complains of difficulty of walking for a couple of months. He also mentions difficulty initiating and performing movement. On examination, the limb shows rigidity on the passive movement. There's no loss of muscle power and deep tendon reflexes. Which of the following marked areas are probably affected?
Number 1
Number 1.
is substantia nigra. Most probably he has Parkinson's disease.
Perfect. MashaAllah عليك. MashaAllah. Allah يوفقك. Perfect. Good job. So it is Parkinson's disease because substantia nigra, as we said, is the dark area because of the dopamine neurons make it dark. So if he has this, if he has a rigid face, stooped posture, difficulty initiating movement, performing new movement, this all indicates basal ganglia issue. And seeing his age, seeing and the other symptoms he have, it really indicates اللي هو substantia nigra being the issue, which is in Parkinson's. Okay. 2 is what? I'm not really sure, but it's clearly, I don't want to like say to myself, I think it's talking about something else, but this dark line over here is clearly marked with the first arrow. As you can see over here, this is clearly the first arrow. Maybe this is indicating that we're inside the medulla. I mean midbrain.
I remember when we reviewed 2 was the red nucleus, if I'm not wrong.
Is it the red nucleus? Isn't the red nucleus on the top? Okay, let me check. Yeah. Yeah, red nucleus is a bit on the top, so I wouldn't say it's red nucleus. I would stick to يعني something else لأن oh, never mind. You're completely right. I don't know why I thought red nucleus was over here, but it's actually pointing at these two dots that are here. I didn't notice. You're completely right. So 40 is red nucleus. It's not here, it's here. And 39 is substantia nigra. So Ahmed, is it clear why it's one, not two? Okay. Okay, now we're gonna go to the second lab, which is spinal cord and peripheral nerves. Just go ahead and get it done. And after we finish spinal cords and peripheral nerves, I'll give you guys a break عشان brainstem as well. Because brainstem is after. Okay, so this is a histology. This is a reminder of a multipolar neuron. This is how a spinal cord looks like. We said in the middle is gray matter, outside is white matter, which are myelinated axons. This is the dura and arachnoid that are not completely attached to the spinal cord. That's something Dr. Active stressed on, is that the dura and arachnoid of the spinal cord is not completely attached. The pia mater is completely attached. You can see this area where kind of the CSF is probably there. And then we said this is kind of a reminder that we have the motor neurons and the multipolar neurons, and that the bipolar is for the special sensations and the pseudo-unipolar is for the sensory. This is a reminder, kind of a section of how it looks when we zoom in the gray matter. We can see the cell body. This is the nucleus. This over here is the start of an axon, and all around is the dendrites. And all these dots where we have blue arrows is the neuroglia or neuropil, which is a group of fibrous tissues, as a group of fibrous, neuroglia, which is supporting cells, and everything around that's not cell is just a bunch of intertwined axons and dendrites of neurons and process of neuroglia. So it's just like a bunch of يعني axons and dendrites together. For the silver stain, this is how it looks. يعني we have, again, this is the gray matter. This is all the white matter. This is how the dura matter is not completely directly connected. We can see that the CSF is in the middle, kind of, in the empty spaces. Yeah, the central canal is over here, and inside the central canal, obviously, you have ependymal cells that create the CSF for the surrounding area. And this is the silver staining of the nerve body of the gray matter here. And you can see the glial cells all around it. This is a dendrite, and all around it is glial cells and neuropil. This is the nucleus, and the dark spot in the middle is the nucleolus. The layers of the nerves is that you have endoneurium, and then perineurium, and then epineurium. Epineurium is the outermost, perineurium is like in the middle, and then epineurium is each fascicle of axons. This is how it looks here. So you have the epineurium being in the outside. You have the axons, individual fascicles, axons here, and around each fascicle you have a myelin sheath, and you can see their cell body colored in blue. And the perineum is all around it because it's in the kind of the sandwich in the middle. This is kind of the same exact thing. This is the picture from pathology, and you can see this is the myelin sheath around axons. This is the node of Renoir between each myelin sheath in order to increase saltatory conduction, which we already know. This is, again, nodes of Renoir. Okay, this is kind of a basic reminder of the spinal cord. We have 31 pairs of spinal cord segments. We have the cervical region, thoracic region, lumbar region, and sacral region. We have two areas where there's a dilation, and it's where we have the upper limb and where we have the lower limb. So the first dilation is in the cervical region because of the plexus of the upper limb, while the second dilation is in the lumbar region because of the plexus of the lower limb. We also have something called the codus medullaris, which is the end of the spinal cord at the level of L1 and L2. But the dura mater and arachnoid mater extend all the way with the cauda equina until S2, okay? And then phylum terminale, where the dura mater and subarachnoid mater stop, and it's only pia mater, and it's called the phylum terminale, where it's kind of a very thin sheath of going to all the way to the coccyx. So ideally, you would want to do a lumbar puncture between L1 and S2 in order to take the CSF, but not be near the spinal cord, because there's still CSF, but no spinal cord. And under it, where you're under S2, where there's no dura and arachnoid, and it's only pia mater, there won't be a CSF. So also you don't need any CSF sample from there, or you won't be able to obtain it. As I said, we have two protrusions, cervical and lumbar, and we said the protrusions is due to the upper limb, and then the protrusion in the lumbar is due to the lower limb. In terms of how the actual segment looks, as you go down, as you go to the sacral region, you have more white matter and less, you have more gray matter and you have less white matter, because there's less descending and ascending tracks going through it. On the other hand, in the cervical regions, you have a lot of white matter, because all the things going to the brain, all the things coming out of the brain has to go through the cervical segments. On the other hand, the sacral, not all of them, for example, the upper limb needs to go all the way to the sacral. So you have very little white matter and more gray matter in comparison, in condensation. What creates stability in the spinal cord? The ligamentum denticulum, which is basically folds of pia mater. The folds of pia mater help keep the meninges in place and stabilizes the spinal cord in general. We have the disc of the bone, and you have the interventricular disc to make it make you flexible and moving forward, backward, right, and left. And it's called the interventricular disc, and there's two parts to it: nucleus pulpus and annulus fibrosis. And usually what happens when you have this herniation is that the actual annulus breaks, so the nucleus pulpus gets the opportunity to protrude outwards. So this is protruding and this is ripping apart. So in disc herniation or prolapse, in certain conditions, the annulus fibrosis can break, and so the nucleus pulpus would bulge out. This is the spinal segment in term. Like if you're in the cervical segment, you have to count 1 in order to know what the spinal cord you have is. Upper thoracic 1 to 6, you have 2. You have to add 2. Lower thoracic is 3. 10 is 1 to 2. 1 2. Yani basically you have to use this table in order to calculate which nerve is coming out in each level, because it's not actually yani the same. As C1 is not C1. As C2 is not C2, like it's different. Okay, now blood supply of the spinal cord. So basically, you have the aortic arch here, okay? And from the aortic arch, we have something called the common carotid. And from the common carotid, we have the from the aortic arch itself, we have the subclavian, and from the subclavian, we have the vertebral arteries. The vertebral arteries go up and join to create the basilar artery. But when they go up and join, they also create anterior branches and posterior branches. So what basically supplies the spinal cord is the anterior branch of the vertebral artery and the posterior branch of the, anterior branch of the spinal artery and posterior branch of the spinal cord. And these come from vertebral artery. So let me read it again. The two vertebral arteries join together in the anterior part and gives rise to the anterior spinal artery, which supplies 2/3 of the spinal cord. Posteriorly, each vertebral artery gives rise to spinal cord. So there is two posterior spinal arteries which supply posterior 1/3 of the spinal cord. Also, we have extra supply from a T8, which is called by the intersegmental arteries, which has a very, very significant influence on blood supply in the spinal cord. So if they ask what is the blood supply of the spinal cord, you're going to say anterior spinal artery, posterior spinal artery, and intersegmental artery at level T8. Okay, so on the spinal cord, we have, as we said, we have gray matter. And the gray matter is cell bodies that connect you between the brain and the rest of the body. We have four that we really have to know, other than the motor ones that we know. We need substantia gelatinosa. Substantia gelatinosa is over here in the wings, and it's responsible for pain and temperature and touch. Then you have nucleus purpuris, which is responsible over here of two-touch discrimination, vibration, and conscious proprioception. Nucleus purpuris is basically for the dorsal column. Substantia gelatinosa is basically for spinothalamic tract, and dorsal nucleus is for the unconscious proprioception. So basically the spinocerebellar tract. So these are kind of the nuclei that they bind to. So again, substantia gelatinosa is connected to the spinothalamic tract by pain and temperature and touch. And the nucleus proprius is for two-touch discrimination, vibration, and conscious proprioception, same as the dorsal column fibers. Dorsal nucleus or Clark nucleus is for unconscious proprioception, which is the spinocerebellar tract. Intermedial which is over here, the dorsal nucleus. The intermedial nucleus gives rise to the preganglionic sympathetic fibers, meaning you only have this protrusion, this green protrusion over here in the preganglionic sympathetic fibers, okay? You only have it at T1 to L2, where you actually have sympathetic fibers or preganglionic. So this protrusion is very unique to T1 and T12, to L2, sorry, T1 to L2. And if you see this protrusion, you just have to know where you are in the spinal segment, because you have the intromedial lateral nucleus, which is responsible of the preganglionic sympathetic fibers. And then we have motor, which we can divide into 3 groups, but we're not going to talk much about this because we already know it from the from the spinothalamic tract. Corticospinal tract. Okay. So just kida for also information about the fibers themselves. So we talked about the nuclei, which is the gray matter. Now we're going to talk about the white matter and where they're located. So the dorsal column, which is connected to the nucleus proprius, which binds to it, is over here, dorsal column. And you have, as you go medially, you have sacral segments, and as you go laterally, you have cervical segments. So things, if you have injury to the cervical region, you have paralysis, you have loss of pressure, vibration, fine tensor, and conscious proprioception in your neck and upper limb. But if you have a problem in the medial aspect of the dorsal column, you would have problems in your lower limb, like like loss of pressure in your legs and your toes, and so on. Then we have another one, which is the lateral spinothalamic and anterior spinothalamic tract, responsible for pain and temperature, crude touch, and pressure. And as you go more medially, it's cervical. As you go more laterally, it's sacral. So it's completely the opposite of the dorsal column, meaning if you have an injury to the lateral aspect of the lateral spinothalamic tract, you will have issue with the lower limb. While if you have a problem with the medial part of the spinothalamic tract, you would have a problem with the cervical region. So you would have loss of pain and temperature in your neck, for example. Not your neck, sorry, your upper limb. The descending tract is the corticospinal tract, which is similar to the lateral spinothalamic tract in the sense that the cervical are more medial, the sacral is more lateral fibers. And that's pretty much it for the tracts. Now just a few pictures that you have to know. So imagine you're at the level of the medulla still, and you have these two before the basilar artery is formed. These are the vertebral arteries. And when they get combined, they can create the basilar artery. This is the phylum terminale. The phylum terminale is, we said like the horseshoe where it's just a bunch of nerves covered by the pia mater, and it starts from the conus medullaris until the coccyx. So now we're off to questions of the spinal cord. After we do the questions, we're going to take a break and then خلاص يعني سهلت الأمور. Things are much shorter now. We're kind of done with the bulk of the PAL session. So can you guys interact with me and let's get the questions done. And actually يعني I noticed in the OSPE there's a lot of spinal cord questions. A lot. Most of the OSPE is like spinal cord and brainstem. So يعني let's perfect our كده knowledge in it. On sensory examination, patient loss of pain and temperature sensation in his left lower limb. Which of the label tracks provided transmits pain and temperature? Mention two arteries that supply this structure.
Is it the lateral spinothalamic tract?
Okay, so where is it? Can you tell me where it is in the picture? Which letter?
I and F. And the
So
artery is spinal artery.
So you're right, it's I or F, but in the question it says it's left lower limb. So what is B? What is it?
The right side, so I.
Yes. So you and Ahmed are right. So it's I, it's contralateral loss of pain and temperature because there is a decussation at the level of the spinal cord. Which are the two arteries that supply it again? Spinal cord. Okay, so the arteries that supply it, as we said, the anterior spinal artery, posterior spinal artery. Anterior is the 2/3 of the spinal cord, and posterior spinal artery is the 1/3 of the spinal cord. And at the level of T8, we have the intersegmental artery. Okay. Yeah, so they are roots from the vertebral artery, but technically the two arteries that supply the structure itself is the spinal artery, but they do come from the vertebral artery, you're right. And this is the answer why. Okay, so I'm reminding you why this is the answer. Which of the following locations would be ideal to perform a lumbar puncture? We said يعني we end at the conus medullaris. We don't want to harm the spinal cord. We said we don't want it where it's only pia mater, which is like at the coccyx level. So what would be the middle ground? Obviously, it's going to be B, and the space between it where you take where exactly L3, L4, and the space where you actually take the CSF is the subarachnoid space. Okay. And we said the conus, as I said, like conus medullaris ends here. We don't want the spinal cord. We don't want phylum terminale because it's where the pia mater is. So ideally, you'd have it in the kind of the cauda equina area at L3, L4. And what is the circled area? The circled area here is substantia gelatinosa. And what is its function? It's associated with the dorsal column, I think, so pain, pressure, and two-thirds discriminant. Sorry, it's not associated with, it's associated with the spinothalamic tract, which is pain, temperature, crude touch, and pressure sensation. So as we said here, substantia gelatinosa is spinal cord, pain and temperature and touch, while nucleus proprius is for the dorsal column stuff. Dorsal nucleus is for the spinal cerebellar tract. What is this, guys? Histologically identify the structure. Okay, this is the nodes of Ranvier, and they increase saltatory conduction. The sodium channels allowing saltatory conduction. Okay. A 44-year-old lady with a 5-year-old history of lumbar back pain presenting with the emergency department with severe sharp pain, now radiating into both her buttocks. She is complaining of urinary incontinence on examination. So she has severe pain radiating to her buttocks and she has urinary incontinence. On examination, the motor strength of her left leg are decreased. So the motor strength is decreased in her lower limb. And the rectal examination, loss of sensation in the perianal region, reduced tone. Which of the marked areas are affected? Yeah, you're right. So it is the caudate equina syndrome. طيب, a 45-year-old hypersensitivity patient presents with sudden freezing pain sensation in her left arm. In addition, she's complaining of weakness and numbness down her whole left face. When examination locally, she was weak and numb on the left side of the chorea of the left arm. Which structure marked is affected in this case? Um, Wait, is this the answer now ولا قبل دقيقة? Did I mess up the before? Oh, it's C. Sorry. Uh, yes, it's C because obviously it's not where the ilium is. Because I lost a bunch of nerves. Any answer for this? Okay, so here it's the thalamus. When you see a bunch of كده random things together, like weakness, numbness, difficulty walking, كده أشياء, pain sensation, you would think of the thalamus. Which of the following neurons are marked or involved in the case of poliomyelitis? Poliomyelitis is did I not put an answer for this? Oops, I didn't put an answer for this, but I think you're right. Yeah, it's actually the anterior, so it would be C because polio it affects the anterior gray horn, so it's lower motor neuron. So C, this area. Sorry, A. Sorry, it's A because this is the front and this is the back. This is the front and this is the back, and this is the front. Okay, so it's A, right? Anterior gray horn. Anterior is here. This is dorsal. Makes sense, guys, صح? Yeah, okay. So patient suffers with penetrating knife injury at T1 level on the left side. Which of the following images show loss of pain and sun temperature on the body, on his body? Last one on which side? Yeah, it's contralateral, so it's over here, this one. Because at T2, when you have contralateral loss of pain and sensation, so they penetrated on the left side, so on the right side you would have loss of pain and temperature. Okay. Good job, guys. Good job. We're gonna take a 15-minute break, or I'll get back to it. Although we need a mental break after this. Hamid, you can. Okay. I just want to say thank you so much for staying. يعني خاسرين فيها ليلة العيد بس لعل فيها أجر ولعل فيها خيرة وبركة إن شاء الله بدراستنا ووقتنا. لا تشيلون هم للي يعني they have FOMO from their family. بس it's okay. أنا راضية وإن شاء الله أنتم راضيين ومعليش يعني نطول عليكم بس يعني إن شاء الله يفيدكم on the long run. بس yeah. So now we're off to the brainstem. There's a lot of cross sections here, but في تكرار كثير. There's a lot of repetition from what we had talked about in the organization of the nervous system. Inshallah, we're gonna go a bit faster here. Just a bit of anatomy in terms of the brainstem. So as we know, we have the medulla, we have the pons, and we have the midbrain. In the medulla, okay, if we really start, we have a medial sulcus over here, okay? This medial sulcus of the medulla. And then next to it, we have pyramids. And then we have the anterior medial sulcus. Let's go take it step by step. In the anterior sulcus, we have the two pyramids next to it, which is the corticospinal tract. So the corticospinal tract are what make the two pyramids next to the median sulcus. And in the lower parts of the medulla, which is here in the X sign, here, it's actually where the motor decussation happens. So at this level, where we become contralateral. Above it, it's contralateral. Anything under it, any injury under it is ipsilateral because the decussation happens at the lower part of the medulla in the pyramids. So again, this is the brainstem. We have the medulla, we have the pons, and we have the midbrain. If we focus on the medulla, we have a line in the middle called the medial sulcus that divide the two pyramids, which are the corticospinal tracts. The corticospinal tracts decussate at the lower part of the medulla. So any injury above is contralateral and any injury under is ipsilateral. Then we have the anterior medial sulcus. Let me add a laser pointer. So we said this is the medial sulcus, and then we have the anterior medial sulcus, which is the color red over here. The anterior medial sulcus comes out, there's another protrusion that comes out of it. It's the inferior olivary nucleus, but that's not what's relevant to us. What's really relevant is that in the anterior medial sulcus, we have the 12th cranial nerve, which is called the hypoglossal nerve. What is the function of the 12th cranial nerve? It's purely motor and it supplies the muscle of the tongue. So it moves the muscle right and left. It's purely motor, and this is what the symbol signifies. If you have an injury at the level of, at this specific nerve, you would have a problem with lateralization. Basically, your tongue will deviate to the side of the lesion, to the side of the actual hypoglossal nerve, okay? So till now, we talked about the decussation. So if we have an injury, for example, in the anterior aspect, you would lose the corticospinal tract in the anterior, and you would also lose the hypoglossal nerve because they're still in the anterior aspect of the medulla. So as we see here, medial portion of the medulla is affected. It includes the pyramids and the anterior medial sulcus. So you would have contralateral spastic paralysis, and the tongue will deviate towards the side of the lesion because of the hypoglossal nerve. Okay? Now, if we go more lateral, now we're more in the lateral aspect of the medulla, we have another sulcus called the anterior, still in the anterior, lateral, not anterior medial, anterior lateral sulcus. And in the anterior lateral sulcus, which is actually the green color that you can't really see clearly here, where we have the arrows as well, I'm pointing at them, okay? We have 3 nerves come out of it, 9, 10, and 11, okay? What is 9? It's glossopharyngeal. What is 10? It is vagus. And what is 11? It's accessory nerve. The glossopharyngeal is sensory, posterior 1/3 of the tongue, so it's the blue part here. And the stylopharyngeus muscle, which is over here, this muscle. And it's a secretory motor for the parotid gland. So also the parotid gland is supplied by the glossopharyngeal. And the vagus is responsible of small part of the ear and the root of the tongue, okay? And motor for the pharynx and the larynx. So dysphagia and dysarthria would be associated with the vagus. And it's responsible of some parasympathetic of the lungs and the heart and the intestine up to the 2/3 of the transverse colon. Then we have the accessory nerve. The accessory nerve has a cranial part and a spinal part. The cranial nerve is responsible and joins the vagus when supplying the muscles of the pharynx and the larynx. So it's, if you have dysphagia and dysarthria, you would have a mix of both. And in the spinal component, it goes to the spinal cord and actually supplies the muscles called the sternocleidomastoid and the trapezius, and it joins the accessory nerve over here. This is what it supplies, the spinal segment of the accessory. And then we have, now we finished from the medulla. Can someone give me a quick revision of what we talked about in the medulla? ما في غيركم أحمد أو محمد. خلاص, straight. I'm gonna talk to you guys. Any recap كذا, medial sulcus كذا, anything? ولا أكمل خلاص؟
What was the question?
Oh, لا خلاص, hey, you were interested. I'm saying a recap of what I just said. Okay. So I'm gonna say a really quick recap. We said we have the medial sulcus, we have the anterior medial sulcus, and we have the anterior lateral sulcus. The anterior medial sulcus, we have the hypoglossal nerve, and the anterior lateral sulcus, we have 3 nerves, 9, 10, and 11. And then we talk about the pons. The pons, what's something we have to know about it is that it has the basilar groove. And in the basilar groove, what do we have? The basilar artery. Before that, if we show a cross section here, what would we see? We would see the vertebral arteries because it's before the basilar artery is formed over here. Okay, and then, so yeah, this is the basilar groove of the pons. And then we have the bulbopontine sulcus. The bulbopontine sulcus is the sulcus that connects between the medulla and the pons. So it's this segment where we have these 2 arrows pointing at it, this sulcus. Okay, there are the there is the 6th cranial nerve, 7th cranial nerve, and 8th cranial nerve. The 6th cranial nerve is only motor. It's for the lateral rectus muscle. Then you have the facial nerve, which is mixed sensory and motor. So sensory for the anterior 2/3 of the tongue, which is over here, and the muscles of the facial expressions. So all muscles of the facial expressions, and parasympathetic, so submandibular, sublingual, and lacrimal glands. Then we have the last one, which is the vestibulocochlear, which is the 8th nerve or the most lateral nerve compared to the abducent, which is most medial, and it's for balance and hearing. Then we have in the upper part of the pons, we have the trigeminal coming out. The trigeminal comes out kind of from the sides, and it is responsible for all sensations of the head and neck, all general sensations of the head and neck, all muscles of mastications for mastica, temporalis, medial thyroid, and lateral thyroid muscle. And it has 3 parts, ophthalmic, maxillary, and mandibular parts. Right, now we're done with the pons. Now we're gonna go up to the medulla. In the medulla, we have we have white matter bundles that are joining at the brainstem with the cerebrum. We have the corticospinal tract and we have the corticonuclear tract, which is the cortex, the cranial nuclei. And we have the corticopontine fibers. So we have a lot of fibers here. And we also have the substantia nigra in the midbrain. And then if we go into the medial aspect of these, so over here, these 2 long things are the corticospinal tract, corticonuclear, and corticopontine. It creates this triangular like faragh, this empty place. And this is where the oculomotor nerve comes out. It kind of comes lying medially and it goes out to the front and lying in the intrapendicular fossa. All muscles of the eye are supplied by the ocular motor nerve, except the lateral rectus muscle supplied by cranial nerve number 6 and the superior oblique muscle supplied by the 4th cranial nerve. And then last thing, in the interpendicular fossa, you will see the pituitary stalk, the mammillary bodies, and the optic tracts curling. So this is kind of like a reminder of brainstem as a whole. This is all from the anterior aspect. From the posterior aspect, what you have to know is that we have the superior colliculus and the inferior colliculus. The superior colliculus for auditory reflexes, superior colliculus is for visual reflexes, inferior colliculus is for auditory reflexes. Why? Because your eye is above your ear. So superior is for eye movement or eye reflexes. Inferior is for auditory reflexes. Okay, this is a reminder of like the orientation of what we just talked about. I'm going to talk about it again. Number one, we said if we start from the pons, we said we have the medial sulcus. And in the medial sulcus, we have the decussation of the corticospinal tract, as we can see over here. And then we have the anterior medial sulcus, which we have the hypoglossal nerve. This is the hypoglossal nerve. If we look here, this is the hypoglossal 12 cranial nerve. Then we have the anterior lateral sulcus, which is over here. And we have three nerves coming out of it. We have the accessory nerve, we have the vagus nerve, and we have the glossopharyngeal nerve, okay? So we have 9, 10, and 11. Then on the bulbopontine sulcus, the bulbopontine sulcus, which is over here, exactly where the line is, we have three nerves coming out, 6, 7, and 8. 6 being the abducent nerve, 7 being the facial nerve, and 8 being the vestibulocochlear nerve. And then over here, we have the trigeminal nerve, coming from the mid-level of the pons. And then over here, in the base, we have the basilar groove where we have the basilar artery. Then we have the cerebral, the peduncles, which are formed from these tracks. I don't want to make them up because I forgot them. Corticospinal, corticonuclear, and corticopontine is what makes these two long green things. And then over here, we have the oculomotor nerve, which is coming medially from the intraventricular foramen. And then 2 coming out from here, but it's not ocular motor coming out from here. This is kind of what they would bring you in the exam to tell you, oh, what nerve is here and so on. So this is just, this is where you're going to test yourself. We said superior colliculus over here is for visual reflexes. Inferior colliculus for auditory reflexes. And then from anterior, we talked about the nerves multiple times, so I'm not going to repeat them. These are the functions kind of summarized in another way and where they exit from. Okay, this is the blood supply. I'm going to go over it. So basically over here, we have the 2 vertebral arteries. When the 2 vertebral arteries, they also give out the anterior spinal artery and the posterior spinal artery, which we said supply the spinal cord. The anterior spinal artery supplies the 2/3 of the spinal cord, and the posterior supplies 1/3 of the spinal cord. And then we combine together and create the basilar artery, which passes on the basilar groove of the pons. And then the basilar artery divides and creates the posterior cerebral artery. But it also gives a small branch called the superior cerebellar artery and gives a branch down called the anterior inferior cerebellar artery. So as it goes up on the pons, it gives small things, small arteries to the cerebellum. And then when it reaches to the level where it's in the brain, it gives the posterior cerebral artery. And then it also gives posterior communicating artery. Then the internal carotid artery comes in play and it turns into the medial cerebral artery. And it also gives the anterior cerebral artery and the anterior communicating artery, creating the circle of Willis. Okay, so let's see about their function. So in terms of the medulla, it's supplied medially by something and laterally by something else. The medial part of the medulla is supplied by the anterior spinal artery and the vertebral artery, both of them, dual supply. The lateral part of the medulla is supplied by the posterior inferior cerebellar artery. The lateral parts of the pons is supplied by the anterior inferior cerebellar artery. So this is posterior, this is anterior. Then the upper part of the pons is supplied by the superior cerebellar artery. Then we have the midbrain and occipital lobe is fully supplied by the posterior cerebral artery. So if you have occlusion of the vertebral artery or anterior spinal artery, you would have problem with the medial medulla. If you have loss of posterior inferior cerebellar artery, you would have lateral medulla syndrome. If you have loss of anterior inferior cerebellar artery, you would have lateral pons problem. If you have upper lateral pons, which is kind of like a trigeminal issue, you would have superior cerebellar artery issue. And if you have a problem in the midbrain or the occipital, you would have the posterior cerebral artery occlusion. Okay? This is a similar picture of what we just talked about. I'm going to go over it again. So over here, we have the two vertebral arteries, and they create this anterior and posterior spinal artery. Then it turns into the spinal, into the basal artery. And while it is going up as the basal artery, it gives off the cerebellar artery. So posterior cerebellar artery, anterior cerebellar artery, inferior and superior. So a bunch of cerebellar arteries. And then it goes and supplies the pons. And then it goes up and divides and becomes, at the level of the medulla, it turns into the posterior cerebral artery, supplies the occipital lobe and the midbrain as a whole. This is something we already talked about, and I'm going to revise again. So second cranial nerve comes out of the cryptiform canal, optic canal. Then we have the superior orbital fissure, comes three things out of it. Cranial nerve number 3, 4, and 5, and 4, 5, and 3, 4, and 6, plus the first branch of trigeminal, which is 5. And then the one after is foramen rotundum. Rotundum creates the second, comes out of it is the second part of the trigeminal. And then ovale creates a space for the third part of the trigeminal. And then we have foramen spinosum, which we have the middle meningeal artery. Then over here, this is all the middle fossa. And then the posterior cranial fossa, we have the internal acoustic meatus causes vestibulocochlear nerve and facial nerve. And then we have the jugular foramen here where the jugular vein comes out, plus cranial nerve number 9, 10, and 11. And then the last one, which is the hypoglossal canal, which has the hypoglossal nerve or cranial nerve number 12. And then we have the foramen magnum for the brainstem and cranial nerve number 11, spinal root of the cranial nerve number 11, and the vertebral arteries. So this is a reminder of the sinuses. Over here, we have the superior sagittal sinus. And then we have the convolution of the sinuses where you have a combination of all the sinuses. This is the inferior sagittal sinus that combines at the confluence of sinuses. And then it divides to create the transverse sinuses on each side. And eventually drain in the sagittal sinus and then at the internal carotid vein. Okay, so now we're going to talk about a few of the cross sections. So we talked about this before. First, we have superior sagittal sinus and we have inferior sagittal sinus. Then we have short association and long association fibers, which we talked about before. And then over here, we focus more on, again, superior sagittal sinus, inferior sagittal sinus, and on over here, this area, it's still the corpus callosum from the top. And then here we can see the lateral ventricles very clearly. And we still see the superior sagittal sinus and inferior sagittal sinus. Over here, we can see that this is the meninges, which is the falx cerebri. And in the lateral ventricle, what would we have? The choroid plexus made up of the ependymal cells for CSF production. We have over here, this is the inferior sagittal sign. You mean anterior and posterior, not inferior. Anterior, لا. You mean anterior and posterior, not inferior. You mean about the superior sagittal sinus? About what do you mean? لا, superior, it's just superior sagittal sinus. It's available anteriorly and available posteriorly when you cut it in a section. But it's just one superior sagittal sinus that goes in a U shape. So when you cut it, you can see it from the front and you can see it from the back, but that's only the visual. That's the reality is it's one tube that goes from the top. And it's one superior sagittal sinus, which we talked about, Tina. It's one, but when you cut it from here, it's going to show from the anterior aspect, it's going to show from the posterior aspect. Makes sense? Yes. Okay. Let me go over them. So we said superior sagittal sinus, which is 1B and 1A. And number 2, over here, this is the inferior sagittal sinus. The same one we saw over here, this inferior sagittal sinus, if you cut from here, you will be able to see it here. So you're seeing this aspect here. Okay. Then you have the anterior cerebral artery, number 16. Which is over here. This is anterior cerebral artery. Okay, this is the anterior aspect and this is the posterior aspect for context. So this is the anterior cerebral artery. And then we have the internal the corpus callosum, which is 32. 32, this is the corpus callosum. 32 is the splenium, which is the end of the corpus callosum, and this is the start of the corpus callosum, which is the genu. And again, it's the same concept of the superior sagittal sinus. It's just a U shape going from the top, and you're cutting off to a specific area of the corpus callosum. We said they have the central part of the we have the parts of the lateral ventricles, and then we said, and then we also have the body of the caudate nucleus at 46, just a random concept. I don't think you have to necessarily distinguish it, but it is the caudate nucleus, which is part of the caudate nucleus. Was it the basal ganglia? Was it which was striatum? Okay. And then we have the this is another cross section. Again, we're repeating the same thing. Here we're anterior, here we're posterior. This is the superior sagittal sinus, and again the superior sagittal sinus is again the splenium of the corpus callosum, and this is again the genu of the corpus callosum. Now we have just new things over here, and what we and over here 30 at 16 over here, we can still see the anterior cerebral artery. We can still see over here, these are the 31, this is the lateral ventricles going kind of to the front now. And you have the thalami at number 15. This is the thalami, and these are the parts of the basal ganglia. And the same applies here. There's really nothing new to check out. Okay, now we're in the upper midbrain. Okay, we're in the upper midbrain. So you can see the optic chiasm over here. You can see the eyes, you can see the nose over here. And over here at this level, at 15, this is the middle cerebral artery that is created from the internal carotid artery. So this was once the internal carotid artery. We still have the anterior cerebral artery. Now we have the middle cerebral artery. And the start of it is the internal carotid artery, because remember, it's a continuation of the internal carotid. So it starts internal carotid, and then it becomes the middle meningeal artery. And then we have straight sinus number 3. We mentioned it as well. What else should I mention? Optic chiasm, optic nerve. I don't think anything else is relevant to us. Maybe just know that this area and this area is the hippocampus. Okay, I didn't see much questions on these specific sections. This is kind of a repetition. I don't think there's anything to add here, but all you have to know is that this is now we're in the lower part of the midbrain and we're starting to see the cerebellar hemisphere. And in front of the lower part of the midbrain, what artery do you think this is? It's the basilar artery because we're almost near the pons and we have the pontine sulcus here. This is the internal carotid artery before it became the middle meningeal artery. Here, it already became the middle meningeal artery. Here, it's still internal carotid artery. Okay. Oh, you know how the vermis is the middle of the cerebellum, sorry. So 58 is the vermis and 59 is the cerebral hemispheres. 3 is the straight sinus. Okay. Number 4 is where you combine the transverse. So this is transverse sinus, this is transverse sinus, and when you combine them together over here in the middle where it's like a pool of veins, it's called the convolence of sinuses. So 4 is the convolence of sinuses. And the 5 is the right transverse sinus, and 6 is the left transverse sinus, and the internal carotid vein is the internal carotid artery is here. The basilar artery is still here because we're at the upper parts of the pons. This is the basilar artery in front of the pons. And what else should I mention? Again, this is the vermis of the cerebellum, and this is the hemispheres of the cerebellum, and this is all obviously the pontine fibers. Okay, now we see the cerebellum more because we're in the lower part of the pons. Again, we're still having the basilar artery at number 18, but what is coming out of the pons over here, number 24, is the vestibulocochlear nerve. At this point, at the lower part, we're not in the middle part of the pons because at the middle part of the pons, it might be trigeminal مثلا. But when you're going to the lower part of the pons, we know we're at the junction of the medulla to the pons. And at the junction of the medulla to the pons, we have vestibulocochlear nerve, cochlear nerve. So that's that. And then number 6, number 9 is it turned into the sigmoid sinus because it's going down. So now, it was the superior sagittal sinus, and then it became the transverse sinus, and then it became the convolence of sinuses, and then it became the sigmoid sinuses on the side because we're going down, so we're kind of draining to the heart. What else should I point out? I think that's all. And then now we're at the level of the medulla. So we're in the upper part of the medulla. 18 again is still basilar artery. We still have the cerebellum. One thing, nucleus in the cerebellum now because of this crooked nucleus shape. And then we have what else? Sigmoid sinuses are here and so on. Okay. I think I'm gonna skim through this as well because we already mentioned the sigmoid sinus is still here, and number 19, anterior spinal artery, because at this point at the lower medulla, this is upper medulla, it's basilar artery. Lower medulla, it's internal carotid artery. Do we have to know 12? 12 is, yes, you should know it because inferior petrosal artery, thank you for mentioning it, because inferior petrosal artery is part of the sinuses that were mentioned by Dr. Akif. So if we're at the level of the upper medulla, you'd know that this is the right and left sigmoid sinuses, and then over here you have the inferior petrosal veins, and in the middle you have the internal carotid artery. For your question about how many slides are left, honestly, I don't know, but feel free to leave if you'd like, because it's all recorded and you can feel free to watch it anytime. I know it's very hard to stay for a long time, but neurology is a very long block. It's hard for me to make it short. Yeah, so we talked about this, and now we're off to questions. So now I want you guys to identify this picture. Maybe it's not as clear, so I zoomed it into it, and I want you guys to tell me what 1, 2, 3, and 4 are. So we have this structure, we have the structure in front of it. We have the connecting nerve over here, and what this big large thing here is. Okay, number 1 is basilar. Good job. Pons, yeah. Perfect. And you know it's pons because of the basilar artery. And then 3, cranial nerve number 8, vestibulocochlear. Perfect. The last one is just the cerebellum. That's all. Yeah. The way I would differentiate between this being the vestibulocochlear or trigeminal, because they have the same root of being going out like this, is that the trigeminal fiber would be much thicker than this. So yes, you're correct. It's basilar artery, pons, vestibulocochlear nerve, and then cerebellar hemispheres. And this is basically the same slide that I explained, and this is what they're asking for with all the boxes. The basilar groove being the basilar artery. The function, and then these are the nerves that come out of the bulbopontine sulcus, but specifically when we mentioned the vestibulocochlear, I just want to remind you that it's for balance and hearing. Patient has having dysphagia in this arteria. Which nerve is being damaged? Specifically in the graph, and the artery supply of this area. Okay, let me do it. So the answer is A. It's over here. Okay, and it's probably the vagus nerve. So it's cranial nerve number 9, if I'm mistaken. Cranial nerve number 10, and it's supplied by the posterior inferior cerebellar artery, because we're still not at the level of the pons. We're still in the medulla, level of the medulla, lateral medulla. So it's supplied by one of the arteries that supply the cerebellum as well, which is the lateral medulla. So over here in the picture, I mentioned that the lateral medulla is supplied by the posterior inferior cerebellar artery. And just a reminder that the vagus nerves is over here. And the function of the vagus nerve is that it's mainly motor for all the muscles of the pharynx and the larynx, and it's sensory for the root of the tongue, and parasympathetic for the lung, heart, intestine, and 2/3 of the transverse colon. Accessory nerve might be damaged as well for the pharynx and the larynx because it supplies it, but I guess the main answer would be vagus. Identify the nerve on the model, abducent nerve on the model. Abducent. So cranial nerve number 6, which one is it? Which letter? So abducent nerve is number D, because you have 6, 7, and then 8. So 6, 7, 8. 6 being the most medial and 8 being the most lateral. So this is abducent nerve being the most medial. The function of the abducent is it's a lateral rectus muscle responsible of moving the eye laterally. Okay. A patient's history of difficulty of swallowing. On examination, there is a loss of sensation of posterior 1/3 of the tongue. Which of the following labeled nerves are affected? Okay, so the answer is glossopharyngeal nerve, typically because of the loss of sensation in the posterior 1/3 of the tongue. And the glossopharyngeal nerve is this nerve over here. And again, oh, sorry, this isn't edited, but the glossopharyngeal nerve is over here. Okay, glossopharyngeal, we said posterior 1/3 of the tongue and the pharynx. Stylopharyngeal muscles of the pharynx as well. That's why he had difficulty swallowing. Now we're off to cerebellum. And so the cerebellum has anatomical parts and physiological parts and have nuclei. Just a reminder that the physiological parts or the physiology is that this hemisphere is for the cerebrocerebellum. So information that are going from the cerebral cortex to the cerebellum. And over here is from the spinal cord to the cerebellum, the middle. And the bottom is vestibular cerebellum. So it's from the kind of the vestibular system to the cerebellum. The nuclei are the vestigial nuclei, the lobus pallidus, and the umbiliform nucleus and the dentate nucleus. The function is that the nucleus vestigii, which is the most medial, are responsible for maintaining the truncal balance, and the interposed nuclei, which is the mix of globus nucleus and the umbiliform, is for correcting movement of the limb, especially in the lower limb. And then the dentate nucleus is for correction of the planned skilled end movements. The histology of the cerebellum is that they have deep folds. And you can see that it has a molecular layer over here. You have a granular layer, and we have a Purkinje cell layer, which is the letter P over here. M is for the medulla, which is composed of all the white matter. And over here, it's basically a mini brain, but it's also covered by pia mater, so you can see the lining of the pia mater here. And it's very deep fold, so it has higher surface area than the actual cerebrum. The medulla is just white matter, so it's the axons are inwards and the gray matter is outwards. This is the layers. We have granule layer, molecular layers, and Purkinje layer, and the Purkinje cell bodies over here that we already mentioned as multipolar neurons. This is another picture of their Purkinje layer cells or Purkinje cells. Over here is the granular layer, and over here is the molecular layer. This is the dendrites of the Purkinje cells. This is also the Purkinje cells where you have them in the base, and then you have the dendrites on the top, and and then you have the granular area here, and on the top you would have the molecular layer. So الحين questions حق cerebellum. Name the connection of the nucleus at X. So what can we see? Which nucleus can you see over here? The C-shaped nucleus in the cerebellum. What would you conclude? So it's the dentate nucleus associated with the cerebrospinal cerebellum, and its function is to play a role in coordination of activity movement, like riding a bike and so on. So as a reminder, we're talking about the dentate nucleus that looks like a C shape over here, and it's related to the cerebrocerebellum, and it's important in correction of planned movement, skills and movements. So it's important in the correcting of planned skilled movements like biking and so on. Off to the limbic system. The limbic system, we talk more about the cerebrum as a whole. So we talk about, the central gyrus, the central sulcus between the frontal and parietal lobe, and the parieto occipital between the parietal and occipital lobe, the lateral sulcus between the temporal lobe and the parietal lobe. And between the two hemispheres, we have the falx cerebri separating the right hemisphere from the left hemisphere. We have the central sulcus, and then we have the precentral and the postcentral. The precentral being the primary motor gyrus, and the postcentral being the sensory. Over here, we have a random area called the Broca's area for language, and then we have the temporal area, which is auditory area. Behind the central gyrus, we have the postcentral gyrus, which is the primary somatosensory gyrus of the brain. we have to know two specific areas for language. We have Broca's area and Wernicke's area. For Broca's area, it's production of speech, while Wernicke's area is understanding of speech, because if you lose it, you don't understand speech here, you can't produce speech. So this is Broca's area, this is Wernicke's area. This is the splenium separating the two lateral ventricles. It's like it's a septum pellucidum. And the splenium is part of the cingulate gyrus. What is Wernicke's area? Wernicke's area is to understand speech. Sorry. It's to understand. It's an area of the brain where you actually create meaning out of what you're saying. So it's not only blabber with nonsense. So it's an area in the brain to understand speech. واضح? Okay, so the splenium is part of the corpus callosum that we already talked about, the genu of the corpus callosum that we already talked about. This is the right and left lateral ventricles, and in the middle, you're separated by the septum pellucidum. Now, the history of the cerebrum. We know that there are six layers of cells in the brain. And what you have to know is layer 4 and 5. 4 being the efferent from the thalamus, and 5 being the efferent from upper motor neuron to the spinal cord or the brainstem. This is how it looks. You must know how to recognize the layer, not recognize, but know the layers. So molecular layer, extragranular layer, extrapyramidal layer, internal granular and internal pyramidal layer, and multiform layer. What you have to know is the internal granular and internal pyramidal. Pyramidal being the efferent, granular being the efferent. Section of the rodent cerebral cortex, and basically you can see it's stained with acid fast. You can see the myelin of the medulla below being darker, and the myelin of the cortex being like much less darker than pink, because it's more cell bodies, and here it's more axons in the cerebral cortex. This picture is what we already saw of the pyramidal cells. This is the blood supply that we already talked about and how the circle of Willis works. Now you just have to identify them here, and basically we have the two vertebral arteries here. The basilar artery connects to them, and then we have the basilar artery divides into the posterior cerebral artery, and then the internal carotid artery gives off the medial cerebral artery, and then it also gives the anterior cerebral artery, and this kind of creates the whole picture. If we go from here, we have the aorta, aortic aorta basically, and you get the left carotid artery gives off the vertebral artery. The vertebral artery combines and creates the basilar artery. And then we have the limbic system, which are parts of the limbic system that you have to know is the uncus, the amygdala, the perihippocampus, the hippocampus, the fornix, the thalamus, and the cingulate gyrus. There's not much anatomy to it. Just know the Papez circuit. The Papez circuit starts with the hippocampus. You bring information from the subiculum through the fornix to the mammillary body, from the mammillary body to the anterior thalamic nuclei, from the anterior thalamic nuclei to the cingulate gyrus, and from the cingulate gyrus back to the perihippocampal region, and that's how you consolidate emotional memory. So over here, point at the MCA and mention a branch. So the MCA is actually here. It's this line, and a branch of the middle meningeal artery, anterior cerebral artery is a branch of it. Occlusion of which of the following vessels would result in the loss of sensation and motor function of the lower limb? Anterior choroidal artery. Perfect. That's a better answer. Occlusion of which of the following vessels would result in the loss of sensation and motor function of the lower limb? So the highlight over here is the lower limb, because if we're talking about the cerebrum, the lower limb, we have to remember the homunculus. Anterior cerebral, yes, you're correct. And which one is the anterior cerebral artery in the picture? It's C. So anterior cerebral artery is C in the picture. So the answer is C. And the reason why it's anterior cerebral artery is because in the homunculus, the lower limb is kind of inside here in the brain, between the cerebral hemispheres. And this is supplied by the anterior cerebral artery. This green area is supplied by the middle meningeal artery, middle cerebral artery. So if you have loss of upper limb, you would have middle meningeal artery. If you have lower limb, you would have loss of B is the middle or D is posterior. Wait. No, the answer is for wait. Anterior and wait, what was the question again? B is middle. Oh yes, B is middle. D is posterior, yes. No, I think E is posterior, D is posterior communicating. But I'm not 100% sure. As we said, يعني the homunculus in both sensory and motor of the lower limb are supplied by the anterior cerebral artery, and the upper limb are supplied by the middle meningeal artery, which is the middle cerebral artery, which is in the screen color. And this is again another picture. The light whitish color is middle cerebral artery. The blue is anterior cerebral artery, and anything below is posterior cerebral artery. And this is a reminder of like the circle of Willis I talked about. Questions. After a patient visits speech therapy clinic with a major impairment of language compression, although speech retains natural sounding rhythm and normal syntax, it's meaningless. Which one of the labeled cortices areas is affected in this case? So it's meaningless. Although speech sounds normal, it's meaningless. Which area? So the area is C, and it's supplied by the middle cerebral artery. So it's Broca's area due to production of speech. Okay. In here. Last anatomy lab before we do micro and be done. So the ventricular system, it's reminder of the steps. So we start with the two lateral ventricles, and then I'll go here. Yeah, two lateral ventricles, and then the foramen of Monro, third ventricle, aqueduct of Sylvius, fourth ventricle, foramen of Luschka, and foramen of Magendie, and subarachnoid space, and then superior sagittal sinus through the arachnoid granulations. So this is another picture that's clear. This is the lateral ventricles. It has a body, it has a horn, it has a it's a very long كده structure, and then we have the interventricular foramen, third ventricle, and then you have cerebral aqueduct, and then you have the fourth ventricle, and then you have the foramen of Luschka and Magendie. And what lines the ventricular systems for CSF production? The ependymal cells. These are ependymal cells. This is the arachnoid granulations that actually sends the subarachnoid space CSF back and drain it to the venous system. And if there's any issue with the granulation where it's blocked or something, you would have hydrocephalus because it wouldn't get drained. This is a reminder of conus medullaris and the subarachnoid space over here for a lumbar puncture at L3, L4, and the filum terminale here where there's actually no CSF, so it's no use. This is a reminder of the falx cerebri, and then tentorium cerebelli. There's a reminder of the venous drainage. We have superior sagittal sinus, we have inferior sagittal sinus, we have straight sinus that combine the two, and then we have the go right and left for transverse sinuses over here, and then eventually they become the sigmoid sinus. You see how it goes down, and from the sigmoid sinus it goes and drains at the inferior inferior something carotid artery. What is it? Carotid vein. Internal carotid vein. It drains into the internal carotid vein. The superior and inferior petrosal artery as well drain directly to the sigmoid, don't drain to the superior sagittal, and so it directly drains into the sigmoid sinus. So again, this is another picture. This is superior sagittal, this is inferior sagittal, this is straight sinus, this is the transverse sinus, and then we have the sigmoid sinus going down. It's not in the picture. This is all the same thing. The lateral ventricle has 3 parts: anterior horn, posterior horn, and inferior horn. This is the same sinus as I mentioned. Notice how the sigmoid artery goes down like this to the internal jugular vein. We saw this picture a lot before. We saw this. This is the straight sinus over here, and this is the superior sinus, sagittal sinus. This is the transverse sinus when we have 2 split parts. This is the transverse sinus again. This is the superior petrosal sinus when it's thin, and then it drains into the sigmoid sinus over here in the circle, which we already mentioned before as well. These are the sigmoid sinuses when you see this fold becomes the internal jugular vein. The inferior petrosal sinus on both sides, but did not show on anatomage clearly, but it's somewhere here, but I don't think they'd ask from an unclear picture. So now this question about identification. So this would be which aqueduct and what ventricle here? Yeah, so it's fourth and cerebral aqueduct. So exactly, fourth ventricle and cerebral aqueduct of Sylvius. Identify A. A is being the straight sinus, and B being and then B is the convolutes of sinuses, and what it's important is that it helps collect all the blood and drain it to one place, to the transverse sinus, and then sigmoid sinus, and then internal jugular, right? And then this is again a baby with congenital hydrocephalus, enlarged lateral and third ventricle and small fourth ventricle. Which labeled area will probably be most obstructed? Most likely, it's probably B, which is the cerebral aqueduct. We have microbiology, very short and sweet. We just have to know all CSF happens. It's better at the lateral position instead of the sitting position. This is safer because you can measure the pressure. You have to send CSF to chemistry and microbiology, and also cell count. So you need 3 tubes. You have to transport immediately, and you do not refrigerate because that would most agents of meningitis are deciduous, so you have to store at room temperature or an incubator or a candle jar. You have to see the volume, the appearance, the staining, you have to see blood, all of these characteristics that make you able to diagnose. So for example, some of the cases where you would have CSF that's turbid or normal CSF or zytochromatic CSF, so that would indicate a lot of things just by appearance. No, let's start with strep. Strep is catalase negative, oxygen sensitive, and bile sensitive. So catalase positive meaning it has the catalase hormone, optogen sensitive meaning it's the optogen drug, it's sensitive to it. It dies from the optogen. It's bile soluble meaning it doesn't precipitate in bile, it dissolves in it. It's diplococci gram positive, and it's deciduous, so it requires CO2. Neisseria meningitidis is gram-negative intracellular diplococci found in neutrophils usually. There's oxidative positive, and it is an encapsulated. Then we have Haemophilus influenza, which is kind of the one we talk about the most. It's very, very small, but it's deciduous because it requires complex material, including factor X and factor 10 and factor 5. which is NAD and heme. And without both of them, it basically can't grow. You can use chocolate agar or heated agar in order to grow it because it has both NAD and hemin due to the heating process. Another test we have to do for Haemophilus influenza is satellitism, which is basically putting Staph aureus in the middle, and Staph aureus breaks down blood. When it breaks down blood, it releases heme and it releases factor and NAD, which is factor 10 and factor 5. And so when you put Haemophilus influenza next to it, it grows a lot because of the staphs producing or releasing the factor 5 and 10 after breaking down the RBC. So Haemophilus would be positive growth area, would grow clearly. So as we can see, hemophilia will grow next to Staph aureus because it will break down RBCs, and when it breaks down RBCs, it will release the required elements for H influenza to grow. Another thing you can do is rapid latex slide agglutation test. So basically, you just use antigen, detect the antigen of H influenza present in human. This is an easier test, a quicker test. And the set of antigens include from A till E, but the most common type of antigen for H influenza is type B, most common cause of invasive disease. This we already know it a lot. It's basically talking about how it should look in terms of like the tube that we showed in the beginning, this one, how it should look. So if it's normal, it should be clear. If it's bacterial, it should be turbid. If it's viral, it should be clear, slightly turbid. If it's chronic meningitis, it should be clear or slightly turbid. What's really distinctive is in bacterial, the glucose is very low. And even in chronic meningitis, it should be very low. Normally, it should be normal 2.2 to 3.3. And in viral, it's also normal because viral infections don't feed on glucose, obviously. So again, what's the name of the test? This is satellitism, and the organism shown in A is Haemophilus influenza, in B, it's Staph aureus. Okay, this is a reminder. Gram-negative diplococci, which one? It's 3 because it's Neisseria meningitidis. And then which organism is associated with consumption of raw dairy products during pregnancy? It's number 1, which is the Listeria monocytogenes, which isn't in lab material, but I saw it in questions and it's in the lectures, so I thought I'd add it. Listeria monocytogenes is especially transmitted in contaminated food like raw milk products, and it's a gram-positive, catalase-positive tumbling motility. Now, just a few random 3 physiology questions and we'll be done. Basically, like I didn't know how to explain physiology because the labs didn't have any material to be explained. So I got a few questions and I thought I'd explain why these are the answers. So this is actually part of the lecture. So this OSPE is like not even mentioned in lab. So which area are we put on the electrode? This is the slide. So he basically told us that the front where the nose is called nasion, and the back is called anion, and the middle is CZ. CZ is the middle. Okay. So what he wants us to know, and remember 5 points: the nasion in the front, the anion in the back, the middle is called the vertex or CZ, or center Z. And you have to know like the two preocular points on the sides. This is where you have to know like where to put electrodes. So the answer here, what are the names of the area we put the electrodes on? We would be nasion and anion, and the marked area in the middle, the circle, is the vertex, or also known central Z. For this question, you're basically detecting the nerve simulated at S is the elbow and response recorded at R. Oh, yes, for sure. Go. Do you want me to re-explain this? Is it clear? So what should we focus on? Just know just know these terminology. And just know in this over here is called, in the front, it's called nasion, and in the back, it's called anion, And in the middle, it's called vertex or CZ. This is like identification of the name of the areas you put the electrodes in. And in the ear area, it's called periocular point. This is what he said in transcriptions. Remember the 5 points where you put the electrodes. And the anion is where you have the protrusion of the occipital bone. Is it clear now? Vertex is the middle. It's the center of your head. It's like over here. This is vertex. Okay. No problem. Okay, the nerve is stimulated at S in the elbow and responds recorded at R. So it's 28 meters away, the two electrodes. If the latency is 4 milliseconds, calculate the conduction velocity in meters per second. So first of all, you have to know, they told you the distance and they told you the latency. What do they want? The velocity. They tricked you because they gave you 28 centimeters and they want it in meters. So don't get this confused. And they give you 4 milliseconds and they want you in seconds. So you have to convert this and you have to convert this. And after that, you do this equation. You do distance over height over latencies after changing. So 28 divided by 100 to change it to meters, and 4 milliseconds divided by 1,000 to convert it from milliseconds to seconds. And after you convert it, you divide the distance divided by the difference between latencies that they already provided to you in the questions, and the answer would be 70. So this is the answer for the question. Clear? Okay. Here, what indicates the latency period? A latency is basically the stop period between the stimulus and the contraction. So the answer would be B. It's like the pause between when you put a stimulus and the contraction. So the answer would just be B. Which of the following makes alpha waves disappear? This is something, again, in the lecture and not in the lab. And so what makes alpha is mental math, because alpha is when you have your eyes closed and you're relaxed. So you just have to reiterate on this concept over here, where alpha is the eyes closed, but the subject is relaxed. Beta is when you're awake and alert. Theta is when during sleep of all ages plus awake children. Delta is very deep sleep, and gamma is associated with high mental function, like thinking and solving math problems and so on. And this is how they look in waves. So another question they could give you over here is, she was relaxed and calm and her eyes were closed. While the recording, he asked her to open her eyes. Which EEG tracing frequency will diminish? What will decrease? Okay, so the answer would be B, because B is basically beta. So then she'll have a busy active mind when she gets this EEG. That makes sense. When she opened her eyes and she started to focus with him. But that's all for physiology questions. This is the things I think you should focus on. And I'm done. That's all. Yeah, for sure I can explain it again. So basically here, the alpha waves is available when your eyes are closed. Your beta is when you're awake and alert, and your eyes are open. So when her eyes were closed, and then he said while recording, he asked her to open her eyes. So what happens when you open your eye? What dominates when you open your eyes? Their eyes are open. It's beta. So the answer is beta, and beta this is how it is in the graph. I don't know if it's worded wrong in the sense of like, oh, you could have said focus on the word will diminish. You can argue that, and maybe that's appealable, but يعني, this is the answer that was available out of. Okay. And that's it, guys. I hope it was helpful. or please scan this code for me to get feedback on my work, and for like how to see يعني how it got. You're welcome. وكل عام وانتم بخير, all the best of luck. Muhammad, you can stop recording now.